Bonjour à toutes et tous. Merci.
Good afternoon, everyone. Thank you for having borne with us. And thank you to the interpreters who are with us this afternoon also. I am kicking off this conference with a certain sense of emotion because it was Michelle who took the initiative for organizing this event. She wanted to organize a conference on the basis of the PASTURE study, and of course, given the dramatic circumstances that we're familiar with, her death, those of us who worked together with her, myself and my team, and in fact, I'd like to thank those who were with us, Claude Auchat, Karim Deli, and others who are not with us necessarily, Tini Metz, who organised this conference with her. As I say, she's left us since, so we're all a little bit moved. In this conference, we are going to be talking about the issue of biodiversity and the microbiome. Important issues for Europe, but beyond Europe, for the wider world.
Excusez-moi, il faut que je reprenne la bonne page de mon introduction. Voilà, merci.
There we are, just let me find my notes.
C'est pas mon bon document.
Sorry, not got the right notes there. Just bear with me.
Ok, c'était pas la bonne page. Excusez-moi un peu.
I do apologize once again. I wasn't on the right page there. Anyway, since the 1960s, the frequency of so-called atopic diseases such as pollen allergies, asthma, eczema, or food allergies has doubled every 10 years. 50 years ago, 5% of the population suffered. Today, that number is more than 30%. Perhaps I can just mention an anecdote that Marie-Monique Robin often recounted when in a class, when she asked pupils whether they knew people who had allergies. More than 80% of pupils raised their hands, and she explained that when she was their age, it was not a very frequent situation. According to the World Health Organization projections, this rate could reach 50% of the population by 2050. Food allergies were extremely rare in the 1950s, but the list of allergens has continued to grow. Peanuts, gluten, lactose, nuts, crustaceans. 8% of European children are affected. These diseases weigh heavily on the public purse. Pollen allergy affects 130 million Europeans, an estimated annual cost of 130 billion euros. That's some 1,000 euros per year per patient. And for asthma, the bill amounts to some 20 billion euros. This spectacular increase in the prevalence of allergies was first observed in the cities of industrialized nations, mainly in Europe and the United States, but not in the countries of the South, to the point that it's paradoxically become a marker for development for public health officials. It's also a real enigma. And it's this enigma that the scientists we've gathered around us today have been trying to resolve throughout their careers. And the good news is that they've managed to reconstruct some of the pieces of this puzzle. Their conclusions are valuable because the explosion of allergies and more generally of inflammatory diseases is now reaching the major metropolises of countries in the south. For the first round table, entitled The Hypothesis of Biodiversity, there'll be 2 round tables and then there'll be one last point tackling these issues of the round table from a more political angle. As I said, the first round table is entitled The Hypothesis of Diversity and the Effect of the Farm. So I call on Professors Terry Hattala, who's here on my right, Erika von Mutius, and Dominique Vuitton. I'd like to start by giving the floor to Madame Hattala, your allergy expert, Professor Emeritus, Helsinki University, and you've been doing research into the dizzying increase in asthma and allergies. You published a paper entitled Hypothesis of Biodiversity in Allergic Diseases, which is considered to be the world Allergy Organization's reference document. So how do you develop the biodiversity hypothesis, and what is it?
Good afternoon.
Switch on.
Good afternoon. It's a real pleasure and honor to be here, and warm thanks for the organizers to put this occasion up. I will speak about biodiversity and health. But first, let's have a look at this photo, which is probably the most influential environmental photograph ever taken. It's taken 55 years ago by the astronaut Bill Anders. And that is the first time the human race really saw, there we live. That's what we use. Maybe we are also responsible for it. It's not the sunrise, it's the Earth rise, the blue planet. 2015, Lancet Commission made a new concept, so-called planetary health, which is the health of human civilization and the state of the natural systems on which it depends. Very important concept. Well, I would like also to correct a categorical mistake. We often speak, we are here and nature is there. And in fact, if I look around here, there is no signs of nature in this room. No green walls, no green plants, no, nothing. Nevertheless, we are an inseparable part of nature. So we should rather say we and wider nature. Nature is beautiful, and we are very fond of it in many respects, but first and foremost, it's a battlefield and a survival game. And the human race has been very successful in this survival game. In 150 years, the world population has grown from 800 million to 8 billions, so 10 times more now than 150 years ago. Well, that is thanks for the so-called cultural evolution, which is built on the biological evolution, with all these technical innovations and medical achievements. But the downside, the other side of the coin, is the fast change of environment, and of course we are all speaking about climate change and nature loss. The urbanization is so fast that the United Nations is stating that in 10-15 years' time, 70% of all people on Earth will live in cities. So there is a great disconnection of man from the natural environment, from soil, natural waters and air. And if you look at the children there on the left, that's an urban environment and quite nice. But look at the children on the right side. They're in a natural environment. I think what the children is there, breathing, touching, maybe picking berries and eating, maybe drinking from the spring. And there on the left you see this, a baby who is so well protected that almost ready to fly to the moon. Well, biodiversity was first defined in 1992 by United Nations. It's variety of life on Earth, and it's concerning the genes, the species, and the ecosystems. And the ecosystem diversity, first and foremost, it promotes stability through various mechanisms. Well, let's go to clinical practice. I am basically a clinician, and I started my career by taking care of patients with tuberculosis. But here you see that microbial diversity in practice means a lot of species but small amount. Lot of species but small amount. But then if an inflammation hits the skin, the microbiota collapses during the inflammation, and the opportunistic bacteria like Staphylococcus aureus is taking the dominance. And maybe the patient has to go to a doctor and get an antibiotic to kill the dictator and get corticosteroid ointment to treat the inflammation. So, dear European decision makers, health is democracy, disease is dictatorship. Well, let's go on to eastern part of Finland, which was divided after the Second World War with the Iron Curtain, which meant that on the Soviet side, the small-scale, self-contained agricultural life continued, while on the Finnish side, on Finnish Karelia, the more Western type and urbanized lifestyle started to dominate. So we compared these two areas and first the allergic conditions. And the difference, the disparity was just astonishing. Hay fever pollen allergy was almost non-existent on the Russian Karelia, even though the distance between the research sites was not more than 200 kilometers. Also, food allergy was almost non-existent, and we did not find any child with peanut allergy. Then if you look at the right picture there, those are adults. And if you look at those born in the different cohorts born in 1940s, there's no difference between the Russian and Finnish adults born in 1940s. But then some difference starts to appear in those born in late 1950s. And these studies were done in early 2000. And if we come to those 40 years later who were born in 1980s, the difference is manifold. So if we would understand what is causing this kind of a different development, we might take steps from treatment to prevention. And yes, the microbiota was very, very different. There on the left you see the microbiota on the skin on the Finnish side and then on the Russian side. The difference was very, very significant, both in richness and diversity of the microbiome. And this is dealing with bacteria. And the biodiverse microbiome, it has a very important function there. It is guiding the gene activation and then epigenetic regulation to adapt us to the living environment. Well, then on the Finnish side, we looked more closely the environment of the school children in terms of plants, richness, and land use, and it appeared that biodiversity loss around the surrounding the environment around the home, if we noticed the biodiversity loss, so the environment was naturally poor, it was associated with poor microbiome, and it was associated with immune dysfunction and allergy risk. So, of course, the human genetics predisposes to allergic disorders, but the Finnish and Russian allergy contrast is due to the environmental and lifestyle factors. The human body is an ecosystem. It's an ecosystem, and immune defense is a response to biodiverse environment. And we are protected by two layers of biodiversity, and they are both getting poor. They are in the wider nature, but also in us, in you. We could use the term nature, even nature deficiency. If we go 500 years ago, Leonardo da Vinci was making this drawing of Vitruvian man, which represents his attempt to relate man to nature. With a small twist, the microcosm is related to macrocosm. So we are losing protective factors, the microbes, but also biogenic chemicals. There are more than 1,000 identified. And we have also strong mental and socio-cultural contacts to nature. So we tried to implement these ideas in the so-called Finnish allergy program, and we were endorsing immune tolerance and resilience instead of avoidance. We, of course, would need targeted avoidance still. If you are severely allergic to peanuts, you should avoid it. But nevertheless, more for the tolerance and also more discussion on health and not disease. And there we had the group of healthcare professionals, biologists, scientists, and also, importantly, the patient organizations. And the results were very, very favorable in terms of allergy diets, which went down. The asthma patients, they were marching out of hospitals. The work-related allergies were reduced. And importantly, there were major cost savings. If you look at the yellow bars there on the right side, there was a cumulative saving over the 10-year program, 1.2 billion euros. When the investment for the education of healthcare professionals and lay public was 2 million. With 2 million, you got 1.2 billion. The good news is that also there are controlled testing now, which shows that even in 2 weeks' time, in a placebo-controlled trial, you can increase the microbiome and get influence on the responsibility. And also, even only 2 to 3 hours per week spent in nature increased reporting good health and well-being. There is also a study showing that if young people are shown a video of wild nature, it improves vitality and restoration compared if they are looking at a video of urban environment. Well, the non-communicable diseases seem to share the same kind of characteristics: microbial imbalance, long-term immune dysfunction, and low-grade inflammation. In the Finnish and Russian Karelia, the disparity of diabetes was also very large, 6-fold difference. But we are very, very worried at the moment for depression, of Alzheimer's disease, increases of cancer, inflammatory bowel disease. And indeed, also there is another group in Finland who showed that in 20 years' time, the intestinal microbiota is getting poor. This is a pregnant woman in the city of Turku from 1997 to 2017. So we need whole society with us to make the changes in lifestyle and behavior, in social structures, and we should push up the green economy. And we have tried that by moving forward in the so-called Lahti Regional Health and Environment Program, where we have combined, we have integrated the public health goals and the environmental goals in terms of asthma, diabetes, obesity, and depression. And 2 years now, after 2 years, the first results seems quite favorable. So we should turn the biodiversity hypothesis positive and to test that in control studies and real life. The modern urbanized citizen is suffering from nature deficiency, which is a new term, which should be diagnosed, prevented, and treated. And that may take that into medical practice to use this term. So, thank you so much for listening, and thank you for your interest.
Merci beaucoup.
Thank you very much. Well, I suggest we take the 3 speakers and then we have questions and answers. Next, Erika von Mutus, you're a pediatrician and allergy specialist and a researcher at Ludwig Maximilians University in Munich. You're also the creator of the PASTURE study. Your work focuses on the causes of the development of asthma and allergies in children. In your first study, you compared the prevalence of these diseases in the 1990s in Munich and Leipzig. What did you see? And in the 1990s, what were the assumptions put forward to explain this increase in allergic diseases in Western countries? And what is the hygiene hypothesis? Microphone, please, for the speaker. Thank you very much.
So just to alert you of potential conflicts of interest, but basically I want to start, I'm a pediatrician, and I want to start with asthma. You have heard the high numbers of affected children from allergies. Asthma is, if you go to a school class and you have 10 children, one of these 10 children will have asthma. So you can really count how many these are. And asthma is the most common chronic disorder in childhood. It often starts in infancy, when the child is just a baby, and it often goes on until they become adults, and then it can also go on further into older ages. So it's a very common chronic condition. Overall, the estimate is that there are lots of people, 358 million people are worldwide affected. And for me in my practice, I know these children often from when they come as babies. And I let them go when they're 18 years of age. And we can treat those patients with some side effects, but we can treat them. But more importantly, there is no cure. So once you have the disease, you basically continue with the disease. And therefore, from my point of view, from a pediatric point of view, I think what is really important is to have prevention, and prevention of a disease that goes lifelong and that is the most chronic is certainly important. And so, like Tari Hattala, I have also a project of an observational studies, and these are in these environments. These are little villages in rural areas, and you can see some of these are farmhouses, traditional farmhouses, where these are family-run businesses. Mother and father work in the homes of these children, take care of the cattle, mostly dairy business. And we have seen in a number of studies, PASTURE is just one of them, in a number of studies that these children are protected. Everything where you see a red circle means there is protection. And interestingly, there are really 2 parts of the protection. One part is the allergy. And I'll show you some data more. It protects you from hay fever, asthma, just as what Tare just has shown you. But it also protects you from viral infections. It's a 2-pronged, orchestrated, protective environment. And so if you look at what are the main important factors on these farms, and we have done this over the last 20 years, we have really scrutinized everything that is around, and you can see that the most important is the contact and the cowshed. If these children are taken to these animals in the cowshed, and if they consume their own milk, meaning the raw milk. And you can see, for example, in the first column, this is asthma. It goes, the white column says they don't have these exposures in the first year of life, about 12%, as I said, about 10%, every 10th child is affected, but it goes down to 1.4% if they have both of these exposures. And then you can see that for all of this, hay fever, asthma diagnosis, asthma symptoms, hay fever diagnosis, hay fever symptoms, and then also an allergy test, that's the last. On the right-hand side, you can see tremendous differences. So it's not just about reporting less and reporting less disease at school age, but it also really having less positive allergy tests at school age when in the first year of life you were exposed to this environment. And that's a picture taken from our birth cohort study, the so-called PASTURE study. It's one of our study participants. And this is what we have found to be the best protective environment for asthma and allergies. in our studies. And we have the PASTURE study was a multi-center, European-funded study, and so we have looked at different countries in that study: Austria, Switzerland, France, Germany, Finland, and you can see that these dots, they're all towards the left, meaning that this is a protection and that it is in every single country, in every single population, we have been replicating these findings. And you can see on the right-hand panel where it says exposure to animal sheds, no, or these three hours. You can see we looked at how much time they spend in the cow sheds, less than 2 hours per week or more than 2 hours per week, which is 20 minutes a day. You can see that this point moves to the left, and this means that we have about 60 to 80% reduction in risk of asthma. This is very substantial. This is very substantial. I will briefly touch upon the milk consumption, but Dominique Guiton will do a much better job in explaining this, just to say that we have this effect on a number of outcomes. The first is food allergy, and we had different ways of testing this. This is just to say there's a very important and significant protective effect on food allergy. There is a very protective effect on hay fever, pollen allergy, and there's also, you can see this curve going down, a very significant protection from asthma. The important thing, and I think that Dominique will go into this, is when you heat up the milk, when these farmers boil the milk, that the effect is lost, meaning that probably there are heat-sensitive components in this milk that drive the effect. But I've been working on these European farmers for quite a long time, and then I had the opportunity in the US through a colleague of mine, Dr. Mark Holbreich, who is an allergologist like I am, and he told me the story of the Amish people. And he had observed in his practice that Amish people don't have allergies, and so he asked me to come over and join. So what are Amish people? Amish were people from Switzerland who emigrated to the US in different ways of emigration, and they retained a lifestyle. This is like we had it before World War I, basically, very rural, no machine, no electricity, no electricity. Everything is done by animals, a lot with horses, but it's also dairy farming, so they have cattle. And the children are exposed very early on in these contexts because the cowshed is very adjacent to where they live, and they are highly protected. So we did a first study comparing the Swiss, because we had the Swiss in our studies, comparing to the Amish children, and you can see by yourself in the Amish, these were 138 children, and I did the allergy testing myself, and you can see that there was, I think, one case of hay fever. There was only 7% of positive allergy tests, and those who had these positive allergy tests were not sick. And you can see the reference, the comparison is the Swiss rural but non-farming, and this is up to 44%. So you can see what the extent of the allergic sensitization has taken over in the population. And for asthma, the difference is less marked, but these children don't have asthma as we have it here in our areas. This is more a viral condition. Anyway, then with Carol Ober in Chicago, she studied the Hutterites. The Hutterites have very many environmental exposures, very similar to the Amish, the same type of food, large families, and so on. But they took on industrial farming, and the children live in these colonies, as you can see down there, and they're not exposed to the cowsheds. That's a very important issue. The children are not exposed. And if you compare the Amish and the Hutterites, you can see for yourself. The Amish, a positive allergy test, which is meant by atopy, is 7%. In the Hutterites is 33%. And you can see for asthma yourself. Very, very strong differences. Again, one being the traditional rural farming, lifestyle where the children are exposed very early in life, and the Hutterite, the industrialized, where at most children at the age of 7, and mostly the boys, would start being taken to the cowsheds. So I think the important lesson here is the environment offers protective exposures. And as Tari said, we did not find any difference in the genetic background. We tested for the genetic background, no difference. It's vastly replicated. It's not just in our hands. There are over 40 studies now worldwide that show this farm effect. It's very solid evidence. The important exposures, as far as we could define them, are cowsheds and consumption of this raw milk. And as I said before, all other potential lifestyle factors, and we've really gone through everything we could possibly think of, do not explain it. So what happens in this environment with the children's immune responses? So we measured a lot of cytokines. Cytokines are mediators of inflammation. And you can see that comparing Amish and Hutterites in the Amish, everything is down-regulated. So it's a profound and strong suppression of inflammatory reactions here. And also at the center is the innate immunity. I don't want to go more into this, but maybe this is something that, if you have heard about this, it's the first line of defense against microbes that plays an important role here. I'm an epidemiologist, and epidemiological studies always are accused of not having causal inference. And so therefore, we took this into controlled experiments in animal studies. The first thing was Donato Vercelli took dust, basically collecting dust from the homes of Amish and the homes of Hutterites, because we think this is the environmental sample that is important. And then she took these dust samples and extracted, basically she washed it in water, and the water that contained the substances included in the dust, this is the extract, was given to mice intranasally, according to this protocol. And what she found is the following. If you concentrate on the left-hand side, where you see these curves, the orange curve is an allergic mouse. The airways contract. The gray line on the bottom is the non-allergic mouse. Not much happens. The red line is the Hutterite dust. It makes it even worse. And the blue line is the Amish dust. And the same is for the cells that are important in the airways. And we did the same thing in our European. We didn't collect in the homes, but we collected in the cowsheds. We went in the cowsheds, collected the dust, extracted the dust in water, the same procedure, and then we gave it intranasally to mice. And again, the red line is the allergic mouse. You can see these airways contracting. You can see the control, non-allergic mouse being down there. This is the green line, and the blue line is really if we give these extracts that were from the cowsheds into the nose of the mouse. And the same thing for eosinophils. They're important cells in the asthma pathogenesis, and we can completely block that red bar, boof, down to 0. So we have a proof of principle that this is not just observation, but this works in animal experimental setting, and basically we can prevent allergic asthma in these mice by intranasal application of these extracts. And also the raw milk is based up by experimental studies. This is a study from Susan Abring, where the same thing, you show this line going up, it's the allergic mouse, and then you can reduce it if they drink the raw milk. However, the effect is a little bit, it's reversed when they heat the milk. Same thing as what we saw in the epidemiology. So we can think that these are really strong factors. So what is so good in these environments? And we think it's the diversity, much as what Tari showed you. Look at this. You have animals, you have hay, often you have straw, so you have plants, you have microbes, you have fungi, you have all sorts of biodiverse exposures. And we have seen that is the diversity of this exposure. So to the left, it is the bacteria. The blue line that goes up says the more of these bacteria you have, the more likely you are a farm child. I think that's trivial. But the red line is interesting. That goes down. The more of these bacteria you have, the less risk of asthma. The left panel is for bacteria. The right panel is for fungi. It's pretty much the same pattern. So what has this to do with the microbiome, and in particular the gut microbiome? In the PASTURE study, which is a birth cohort followed from birth now up to the age of 6 and beyond, we sampled feces to look at the gut microbiome, and we sampled the feces at age 2 months and at the age of 12 months. And we know that the microbiome matures. It becomes much more diverse, as you can see by the more colors at the age of 12 months. And that pace, velocity of maturation is important. If there is a more rapid maturation, then you can see again in these 4 countries of the PASTURE study, Austria, Switzerland, Germany, Finland, it all goes towards the left. There is protection from asthma if that gut microbiome matures more rapidly. And if there's this more rapid maturation, this is due to having more siblings, larger families, having exposure to cats, growing up on the farm, exposure to animal sheds, consumption of the farm milk. I mean, this you can say this is the hygiene hypothesis. All of this results in a more rapid, we may also say in a normal maturation of the gut microbiome, whereas in the absence of these exposures, it is delayed, and this delayed maturation is a risk for asthma. And so I think, much as Tari has seen, there is this external world where you have plants and animals and all of the natural environment, and the interface between this environment is the microbiome. And it is on various body surfaces. It can be on the skin. For asthma, it's in the respiratory tract or in the intestine. And these bacteria produce metabolites. They produce substances, and these substances can go into the blood of the host and mediate the effect. And that can be the barrier function, that can be immunity, or even lung function. So in summary, the environment, I think that's the important part here, and I fully agree with Tari that we are part of nature, and the environment provides a natural source of exposures that protect from asthma and allergies. These are beneficial microbes that have been found and that mediate this protection. These microbial exposures result in a more rapid, I would call it a normal development of the gut microbiome. And the most important message here is for all of us that we need to find ways how to restore the lost protective exposures to prevent asthma and allergies. And thank you for your attention.
Merci beaucoup. Alors, Dominique Vuitton.
Thank you very much, Dominique Vuitton. You're an immunologist, member of the French Academy of Medicine. You participated in the PASTURE study in Franche-Comté, dealing with consumption of raw milk by sort of farm children. What did you observe, and what's the role of farms and consumption of raw milk when it comes to prevention of inflammatory diseases?
Thank you. Thank you very much for the introduction. First, Happy New Year to everybody. And thanks to the organizers to have imagined this wonderful conference with the presence of colleagues, international colleagues, who have become friends. So I was asked to speak French, so I will turn to my maternal language, and my PowerPoint presentation is in English. So Erika already, Erika, c'est déjà, non, ça va aller. Erika
already introduced the PASTURE cohort without going into too much detail. So it's what we call a longitudinal study because it follows a population throughout their life. And this is from before birth, because it's in the third trimester of pregnancy that the children were recruited. So 500 in 5 different European countries, born and living on a farm where animals were raised, and 500 who were born in the same rural regions in these same 5 countries, but who don't live on a farm. The children were followed to the age of 17. The last visit took place 2 years ago or a year ago, depending on the COVID pandemic. And I just wanted to stress the wealth of data we were able to obtain from the PASTURE study. We had thousands of questionnaires, also differing samples taken, which allowed us to carry out microbiological studies, genomic and immunological and genomic studies, which is quite rare for cohorts. The children had medical checks at the main steps of development throughout their childhood, the last at 17, as I said. So 20 years we've been working now with this population and families. I was asked to concentrate my comments on the influence of raw milk consumption. The first question is what have we learned from studying this PASTURE cohort as far as raw milk is concerned. The results confirm what previous studies had indicated, and that is notwithstanding any kind of contact with animals or experience in the cowsheds, as Erica mentioned, drinking raw milk protects against the development of allergies later in life. Depending on what the studies we carried out, we were able to demonstrate this with atopic dermatitis, asthma, allergic rhinitis, or a more general allergic sensitization. And as Erica also briefly touched on, we were also able to show, although this wasn't the focus of the PASTURE study, but we were able to show that drinking raw milk protected against respiratory infections in the first year of life. You don't really see it too well on the panel in the middle there, but it shows that there is protection against otitis, which is particularly significant. Now, this raw milk, we got the impression throughout analyzing our data from the PASTURE study was that this was the factor for protecting, because it was the most constant factor we found throughout our analysis. This is notwithstanding any other farm-associated factors. And the mother's consumption of raw milk also protected the child, and the consumption by the child at the beginning of life is also important. But I'll come back to the significance of continuous exposure. We also observed this in the non-farm children. If they drink raw milk, they also have significant protection. There's also an additional effect of farm milk products, whether it's butter, yogurt, or cheeses. On cheeses, our group in France, in Besançon, in cooperation with a researcher from INRAE, Sophie Viclos, looked particularly at this aspect, and we were able to demonstrate that the consumption of a wide variety of cheeses in terms of how they're made early in life significantly protected against the development of atopic dermatitis and food allergies at the age of 6. We were also able to show that children that never or only rarely ate cheese had a higher risk of developing food allergies and also allergic rhinitis. Now, the Finnish children had the highest frequency of cheese consumption. However, France managed to save its honors. It was the small French children who had the broadest range of cheeses that they consumed. But then there's the duration issue of consumption or exposure. Now, dogma would have it that only very early exposure in life, in particular the first year of life or up to 2-3 years of life, that will count. That's the only thing that counts. And that after that, it's too late. But study from using PASTURE data, which was recently published in 2023, showed children, depending on their consumption of raw milk throughout life. So rather early or slightly later, these were children that were checked at 10.5, or around 10.5, and then an ongoing consumption. So you can see that in the left. And what we were able to see is that, in fact, optimal significant protection is achieved with continuous consumption. In addition, as Erica said, that there is more protection rather than just early consumption. So that sort of undermines the dogma of this window of opportunity that we've learned, so that there was only a specific time that we can intervene and no other. Now, obviously, we wonder what is in raw milk. That is the protection factor. Well, the first hypothesis from Alex's study and coordinated by Erica touches on bacterial endotoxins, but we didn't find more endotoxins in raw milk from the farms we studied than we found in milk bought in the corner shop. Now, there was a higher number and diversity of microorganisms in raw milk, and that reflected the farm environment, really. And the more traditional the farm is, the more diverse these microorganisms are. We didn't see an association between the quantities, so the numbers of organisms, or even the presence of a given species, and the clinical parameters, so whether it's an allergy or infection. But we were able to show, and this is something Erica showed as well, was the association of this protection with microbial diversity. And no matter which criteria we found significant in the PASTURE study, this diversity aspect was found to be the key word from the whole study. But in raw milk, we shouldn't forget that there are other components that could have an impact. In particular, we stressed the possible role of whey proteins, also the immunity components that exist, in particular cows that are in pastures and that are in fields with more than 1,000 cows. But there's also lipids present, in particular the conjugated linoleic acids, omega-3s, fatty acids, and short-chain fatty acids. There we have two examples of the PASTURE results, which look at this lipid concentration. We were able to show that milk drunk by non-asthmatic children was richer in omega-3 than the milk drank by children who were asthmatic. And we also studied the concentration in butyrates and propionates in the cells of the children. These are seen as immunomodulator cells, and we did see less allergy sensitivity in those children who were drinking this kind of milk. Obviously, the main problem is determining how this works. So what's the link between the farm environment and drinking raw milk in particular, and the immunity system and the protection against inflammatory diseases? Now, Tarja already mentioned this, and so I won't go into detail on this, but what we can say in a sort of rather rough immunology approach is that during the fetal period, the fetus is protected by having an immunitary environment. You can see this on the left. And if you look right to the right, you see the allergy profile is fairly similar. And that's something that would indicate that in order for the child to be quickly able to fight against the infectious agents while tolerating the proteins and non-dangerous microbes, that there needs to be this rebalancing going on as soon as possible. So there's a series of factors which are in red, which are almost all linked to microbian biodiversity, will lead to a better balance through the T regulatory mechanisms. And then on the top, we have a series of green points, which are factors which would rather increase the allergy risk and would not allow for a regulation of the immunity system. The little diagram on the left sums up. I mean, the PASTURE study is 70 scientific publications, so it's a quick sum up of those results. So if we sum up, we can see how the sequence takes place of the environmental and nutritional factors and the results. If we want to have a rebalance of the immune system, which perhaps goes through predisposition on the genes, so the epigenetics, and then those that are linked to the environment. There's direct paths, indirect paths. So one of the main indirect paths goes through changing the microbiome, as the previous speakers have said. Now, in practice, just to give you a few examples, because what we've seen in the PASTURE study and linked to the consumption of raw milk, we've seen that fetal exposure, so before birth, without any link to the environment, simply through the mother's environment, so there are changes, immunological changes. This is one of the first studies from the PASTURE study, which says the umbilical cord blood, we already saw immunity changes that were heading towards rebalancing this as soon as the children are born. So if we look at the child's exposure now, and this is also associated with immunological changes, and we were able to show actions both on the innate development as well as the adaptive immunity, as we call it. On the upper part, you can see an example of change to the toll-like receptors, and then below, which is difficult to read, but the message is that the creation of T regulatory lymphocytes, this was a study done on a sample of children of the German and French segments of the cohort. So, to finish, what's the role of the microbiota in the development of the immune system? And more particularly in what we've observed. I won't go into too much detail on this because you'll have a whole panel of experts dedicated to this who will tell you everything about the microbiota. And Tari has already mentioned that the microbiota already quite a while ago now. I mean, we saw this in the earlier studies that the microbiota were different from in children living in so-called developed environments compared with more traditional environments, different between children with and without allergies, and are generally more diverse and from in children in a rural area. This was confirmed by numerous cohort studies. For example, the Canadian child cohort, which showed the same differences. Erica basically said everything I wanted to say to finish. That is the results of this study that was done on the microbiome at 2 months and 12 months in the children in the PASTURE cohort. To sum up that, an inadequate maturation of the microbiota often leads to the early appearance, or there is a link between that and asthma development. We saw better maturation for children living on a farm. What I think she didn't say, and that I can add, is that in a study on hay fever, there was a positive correlation between the richness of the gut microbiome and protection against hay fever at 10.5 years. And this is something I mentioned earlier when I talked about continuous exposure. There was also a positive correlation between the richness of the gut microbiome at 12 months and the consumption of raw milk. Now, this idea of optimal maturation was mentioned by Erica already. So, just a reminder of the main results here. That is, optimal maturation of the intestinal microbiome based on the PASTURE studies is associated with a farm environment, early food diversification, which includes farm milk, products from the farm, and eggs from the farm as well. This is the first time that we mention eggs, but that's something that needs to be looked into as well. And it's also associated with initial breastfeeding, fortunately. But we're also able to show that this initial breastfeeding shouldn't last too long, because that would be then working against the optimal maturation in the end. Very fine-grained studies of the microbiome in these children highlighted networks of bacterial generi and fungal agents. So microbiota ecosystems within that can actually react by producing a certain immunomodulatory components such as butyrate or propionate, which, as I showed in the study just before, this was something that wasn't found for hay fever. So there's still a lot of work to be done to shed light on this, because we're still working on this and we need to look further into it. But what I can say is that the history of the study, I mean, it's already 20 years old, but we're still at the beginning of our fine analysis of the microbiota. There will be new techniques developed that can be used. And there's a whole wealth of epidemiological and clinical information that can be used, but also samples that were taken within these studies. So it makes it a very unique study. I mean, if we get enough financing which allows us to continue working on this fantastic cohort, I'm sure that we'll see even more interesting things in future. And to finish, I would like to thank my colleagues from the PASTURE study. It's been a fantastic adventure, both scientific and human adventure. And I'd also like to dedicate this presentation to Jean-Charles Alphin, my colleague who started the French branch of the cohort with me, and who, like Michel Rivasi, left us far too early. And I'd like to wish good luck to our young pediatrician who has picked up the baton, and I hope will continue with this study. So thank you very much.
Merci beaucoup, Dominique.
Thank you very much indeed, Dominique. I forgot to say that Donata Vercelli should have been with us, but there's been a death in her close family. And so at the last minute, she has apologized for her absence. So that means we have some 10 minutes for an exchange to take questions from the floor to our three panelists. Thank you very much indeed for your attention. And thank you for these presentations, which are very clear, very precise. So do you have any questions in the room? Mr. Bitou.
D'abord, merci.
Well, to begin with, thank you. I think these were very clear presentations. I'd like to come back to a subject that was touched upon by you, madam, the comparison between the Hutterites. I mean, I discovered them. I didn't know of their existence before today, and the Amish. And of course, that brings into the picture the sort of the way of farming, this agricultural model. And it led me to two reactions that are formed into questions. It was a study carried out in the European Parliament, proposed by the Commission, of work-related risks to farmers. And it was found that the main risk when it comes to professional diseases or occupational diseases are exposure to pesticides. Exposure to pesticides is one of the greatest occupational hazards. And of course, that brings into the picture the sort of way of farming. Now, in this study, we're talking about children, but I think beyond the farmers, the adults who are using pesticides in the front line when it comes to exposure to pesticides is the issue of their children, who are also sort of exposed. They suffer from pesticide exposure, diseases such as cancers. So I think there are important differences if you look at the agricultural practices using pesticides or those practices that don't use pesticides on the health of rural dwellers, rural inhabitants, where there's greater exposure to pesticides, but also biodiversity in the environment, biodiversity and microbiome, rural areas that are exposed to pesticides. Is that going to lead to a loss of biodiversity and therefore a loss of the effects that you're talking about this afternoon in terms of the protective factors as provided by the microbiome? So if you look at farming practices in these rural environments, in these differing environments, tell us a little bit more about exposure in children. And my second comment concerns milk consumption. You talked about raw milk, boiled milk, if I could put it that way, without making any distinction between type of farming practices. I think Franche-Comté is a good point here. You have herbivores eating grass. It's good, but it's not the majority of most herbivores that produce milk. Most cows are fed on maize and soy. And there are very interesting studies produced by INRAE in France, and they've shown that this food regime in terms of quality of milk does have a major impact in terms of the ratio between omega-3 and omega-6 when it comes to cows that graze. A lot of omega-3, very little omega-6. Omega-3 is an anti-inflammatory. Omega-6 is an inflammatory. And then when you cows with fed on maize and soya have high amounts of omega-6, but little amounts of omega-3 anti-inflammatory, as I was saying. So these are the comments that I wanted to contribute to the debate. And my question would be that above and beyond exposure, or the differences in terms of children living in rural areas and urban children. I don't know, you said, referred to them as developed or evolved, I don't know. But I think one could think in terms of a dichotomy. Children in rural areas, preserved rural environments such as the Amish, or rural environment that's being less and less protected as a result of a greater use of pesticides or extremely hazardous substances, leading to the breakdown of biodiversity and the emergence of foods that are not necessarily of excellent quality. That's really what I wanted to say. Thank you. I don't know whether there are a couple of other questions that we could take. Then we'll give the floor back to our panelist, Claude.
Merci pour les...
Thank you. Thank you for your presentations. These 3 presentations are very interesting. Very interesting presentations indeed. A couple of questions. Firstly, the PASTURE study has given rise to some 60 publications and a number of scientific reviews. So in the field, how did this study go down with the doctors and pediatricians? Has there been any impact on them? Now, with this sort of overly hygienic approach to life, which runs counter to what you seem to be suggesting in the study, or the results of the study, what about that? I mean, is there any way of sort of going back? Can we put people back in this sort of a more natural environment? Is it possible returning to a more natural environment, raw milk, for example? Can we recreate that type of environment, and how long do you think it would take? Now, in terms of industrial farming practices, I mean, it's what the previous speaker was talking about, given our industrial agricultural practices, the more traditional farming practices, the more organic. Would you say that that is better in terms of our health? I mean, that would be important. Were we in a position to state that? And then in terms of conclusions of your research, do they concern only allergies, asthma, or the other diseases or illnesses that are concerned when it comes to immune responses? Any other questions?
I will not remember this question.
Ok, on va...
Oui, je me suis pas mal intéressée à toutes ces questions.
Yes, I looked into some of these questions a lot some years ago, less more recently, but I think what you said confirms that you can obtain these protection factors or protective factors with boiled milk. Certain fatty acids, that would be the case. The fatty acid which would be protective. I mean, where are you on that issue? Because the conclusions are less or more favorable in terms of farming practices, the influence of the pharmaceutical industry. I just wonder where the thinking has reached on that subject.
Eh bien, la première question avec les pesticides.
Well, the first question on pesticides, I think Tari has shown that there's always a risk factor alongside a protection factor. I think in traditional farming, I think the protective factors are so high in terms of what we've seen. If exposure starts at the early stages in life, there is, how can I put it, It's as if you're putting a brake on the risk of allergies. Of course, we have a major problem with modern agriculture. I mean, I'm a pediatrician and immunologist. I think the main question for me is, are children being exposed? We have collected samples in cow sheds. And it's not a question of the number of cows, how big is the cow shed, or the type of cattle, but it's rather whether the child is exposed and at what stage in life is that child exposed. I think that is the main factor. And in terms of what we've seen in rural regions, if you have neighbors some 50 meters away from a farm, there's a protective factor there too. In other words, there are things possibly that are transmitted through the air that will provide protection, or if children walk through a farm, or if neighbors pop in and out, there are lots of contacts, well, that protective factor then is transmitted. So it's the contact really with the child, between the child and the development of the sort of immune system in these environments. It's that affords the protection. And I think we should never forget that when talking about what type of farming would be more or less protective, I think that's what we shouldn't forget. It's the exposure and at what age. Now, raw milk, I mean, that's exactly what Dominique was showing. There's a very strong impact and it's repeated. But I don't think we can sort of make a general recommendation that we should all be drinking raw milk. I'm a pediatrician and I've seen infections in sort of children in intensive care. So I don't think we could make that general recommendation that children should be drinking raw milk. But I think what we need to remember is that nature, how can I put it? Nature teaches us a lot about beneficial exposure. Of course, we've been talking about microbes in terms of pathogens, but I think what we have discovered in recent years is that a lot of microbes that are very beneficial and that are a good thing in terms of our health. And that is something that I think we need to understand. But of course, it's difficult to get that message across. You can't say, well, just go out there and drink some raw milk. I mean, I'm all in favor of biodiversity. There's no question there, but I think in terms of health, we need to establish the link. How do things operate? What does it mean in terms of immune responses? And it's what you were saying. A short fatty acid, for example, can they be taken, or can we work only by isolating sort of short-chain fatty acids? There are pharmaceutical companies now producing bacteriolysates. They're bacteria that have been killed off. There are various ways of doing that, and then they're given to children in these studies. Well, the evidence isn't all there, but it's very encouraging. In other words, one might not need bacteria straight from the farm, but there are other bacteria. They're not pathogenic. They've been killed off even, but they will simulate or stimulate a response from the immune system in such a way that it affords protection. That's one possibility. And another possibility, and I'm sure my colleagues will want to say something about it. What about the metabolites? What about can we replace the bacteria? Do they have to be alive? Do you have to sort of kill your bacteria? What are the risks in terms of such applications? And there we will, of course, require clinical trials, but we're not at that stage yet. I mean, that's what I think is important. I think what we're seeing is extremely encouraging. There are new possibilities opening up in the area of prevention because that's what we're talking about. That's what's important.
Il nous reste 5 minutes juste avant la pause. Dominique, une réponse, et puis après.
5 minutes before the break, Dominique, and then I'll put a last question to Tari.
Moi, je pense qu'il faut.
Well, I think we need to understand that the 5 zones that we studied in the PASTURE study are zones of traditional farming. I mean, everything's relative, of course, because there's no real traditional, but in terms of the habitat and the practices, was slightly mountainous. The cows were in the fields and the alpine regions where milk is known to be richer in omega-3, etc., as we've said. So we are in an optimum situation, I would say, but I don't know to what extent this was done, at least as far as I know, not on a cohort, was to study hyper-mechanized farms. I mean, that's a very different lifestyle, not very organic. I mean, farms with 1,000 cows in barns with mechanical milkers, etc. I'm not entirely sure that would be as protective, because we had detailed studies of the farms, of course, as well, the farmers that were involved. We had the impression that these were the most traditional farms that were, in fact, providing the greatest protection. Comments off mic, I'm afraid. Comments off mic, we can't hear them, I'm afraid. But the families have a lifestyle which are not the same as the American population, for example, similar farms.
Oui, alors.
Now, I'm speaking for France, obviously, because that's what I'm most familiar with. But I think in Finland, the attitude is slightly different. I mean, Tari showed that there's a desire to work for more prevention. And I think the study of the results here is not very widespread in the medical world. We don't know why. I mean, the money devoted to research was looking at city pollution, which worsened asthma symptoms, but very little has been given to study why there's been this increase in the cases of allergies. And it's also true that raw milk has been subject of bad press for reasons that were mentioned. There's also a series of pediatrician professor who are going to go out on the press to say that you're going to kill the children by doing this. Unfortunately, if a child dies because of consuming the wrong E. coli, then that's going to undermine a lot of the efforts. It's very difficult to push the consumption of raw milk because of those few cases. I mean, perhaps, as Eric said, there's other ways of looking at this. Now, obviously, we've never said to people, drink raw milk, because in one of the publications we had an open letter from angry pediatricians saying, what on earth is this to have children drink raw milk? No, we left it open to the families to do what they like. However, within the dairy and cheese-making environment, in a more traditional farming environment, I mean, they're delighted with this. They're delighted to hear that for once we're saying that it's good for people's health. Thank you, Dominique. Well, before concluding, I mean, there'll be a second round. We can have... Sorry, excuse me.
Non, alors, on pouvait pas...
No. I mean, we discussed this many times with our colleagues from other countries. We thought that there was perhaps only in France, really, where we could ask families to distinguish between raw milk and non-raw milk. We didn't do this because we thought it was impossible to get this information. The study on the cheeses we did with INRAE from Dijon, we started with manufacturing manners to judge the diversity. And we ended up with the results that we got, which showed that it was, it did help with the protection. Obviously, within the study, there was a lot of raw milk cheeses. Okay, thank you. Last comment I'll give before giving the floor to Tari. Just to come back to the example of the Russian and Finnish Karelia, because in the prevention program you set up in Finland, 2 hours per week in nature, it doesn't cost very much. Well, these type of programs are not very generalized, given the preventive nature it might have. Why do you think that is?
You got the point. I was just saying that that we should take, for example, the concept of nature deficiency more seriously, because that tells you if we are speaking about nature loss, we think it's in the wider nature, but nature deficiency is telling that something, you are having some defect in your ecosystem, which is called also the human body. Yeah, then we are, in general terms, we of course can argue what kind of farming environment is healthier than another. That is important knowledge. But nevertheless, if we take the public health view, we should think how can we compensate the nature deficiency for the lay public? By maybe changing the diet a bit, eating more vegetables, fruits, roots, exercising and walking and going out more in a natural environment. Think about our housing environment. What kind of a housing environment we are having? Do we have any kind of a gardening or growing some plants by ourselves? Do we have any contact with pets? And all those things. How we are walking or using a bicycle in our city? Can we exercise and do these kind of things which we know are good for public health? So how to compensate the nature deficiency in the modern citizen who has been urbanized and the diet is changed and the housing environment is changed? That's the big issue in the general and lay public level. Thank you very much.
Merci beaucoup.
Thank you very much. Okay, I think we'll take our first break. We'll start again at 3:30 with a second panel of speakers. So I'd like to thank our first round for their very clear and precise presentations. Thank you.
Donc on reprend à 15h30.
Start again at 3:30.
Thank you very much.
Thanks. I think that you got the point. You got the point.
Thank you.
When you said about, because this is a little bit going too much into detail to kind of argue what kind of microbes are good and bad and what are dangerous and not. This is not the real picture in the first question to solve in the world at the moment. It's of course important to know these details, but we cannot give our children raw milk, for example. It's too dangerous at the moment.
Yes.
And first of all, I really like the presentations and the content, and I followed it for a while. Nevertheless, the raw milk discussion, I think, is a good intro then for my talk. But I do think it's this fine balance, right? If you look at life expectancies, development over the years, hygiene has a contribution to, I don't need to tell you about to the public. So the problem is, there is also a perception in some people, it's just as natural as possible and we will live much healthier, which is also not true. Because we know listeria, right, in pregnant women can kill them and in raw milk. and I've seen that, if you hear about such cases from the medical colleagues. So it is a very complex, I don't need to tell you, a very complex discussion. My concern is that people often get then the feeling, we should just go back as unhygienic as possible. And that's also not the answer.
Totally wrong.
Yeah, I know that you go. But transmitting this message in a way that I'm all with you. We need to. I could sign every sentence you said at the end. Right? We need to go more back to nature and still keep what we have learned and gained from it.
Because these are fantastic achievements.
Exactly. But they finding this balance and maybe finding smarter solutions than just drinking raw milk. Right? Finding maybe we can restore it in another way, because the raw milk is probably not the way to restore it.
No, and it's not really.
And how would you do it in a Chinese city?
Is that for general public, and is that a public health issue?
No, and then
have to find
smarter ways here. But it's
which
so
are politically acceptable.
and also health-wise acceptable, because I agree that these are a few cases, but if you have seen children dying from genetic E. coli, right, this family will not accept if you say, these are just a few cases, because these are preventable deaths. So, but we pay a price for the hygiene with the allergies, and I agree. This is certainly true.
Yeah.
And I think it's a communication and a science challenge.
It's a challenge, but it's not. Because during the allergy program in Finland, 10 years, we did not go into very detailed instructions what to do, but improve your nature contact. Think what you eat, think what you breathe, think what you touch, and how you organize your mobility.
I find all this. But do you think allergies go down? Even then, even without raw milk, it would go down substantially.
Yes,
Yeah.
we don't need.
And do you think how important is this? I'm a microbiome researcher. We'll see later. But how important is this window of opportunity for at the very early life stage? So can you still revert it later relatively easily or not?
This is an important question, because if you think about the immune system, the major part of the pyramid is the immune, the first line. Well, what's the point?
Studied in neurology, yeah.
And what's the point also that it does not create permanent memory cells. It must be exposed throughout of life. And that means we were speaking about children all the time, but it's also important for the elderly.
On the other hand, there were data where they showed if you do it later, I think they showed the milk thing, then the effect is much smaller than if you
It's
do it
more. It's more. But when you think about Alzheimer's disease, for example, which is along with urbanization, it's going like this. If you think about depression in young people,
I agree.
if you think about increase in breast cancer and cancer,
No, you don't have to convince me, I'm 100%.
but the point is, here if we just stick to the first year or first 2 years, this is not the solution.
No, it's not. That was my question, exactly. This is not the solution. No.
It should cover the population of all ages, and the European population is aging fast. The elderly also need a parrot there on the shoulder. And of course, it does not have
but it still has a beneficial effect. And often it's even more complex. If you think about multiple sclerosis, now when we know that also the Epstein-Barr virus, right, has quite a contribution. If we would get a vaccination against Epstein-Barr, we could probably prevent all or a lot of the multiple sclerosis cases, independent from hygiene. So they are often, it's here an infection, right? An early life infection is the cause, or at least contributes, is essential for the development of the disease later on. So sometimes it's also interesting.
It's always a question of balance.
Yeah, exactly.
I have a question for you, if it's okay.
Yes, please.
Can you show me just for 1 second how to pronounce this? Yes. Because no one will probably show me. You were sitting here, so how did you do it?
Here. This is this. And you see that there, that there.
Can you
and you
point
can
to,
change.
or you know?
No, you cannot.
You just
You can only change.
You can only move forward.
Then you, well, no, you know these.
Yeah, I don't, thanks.
You will be using this for your next, for your speech.
Yeah.
Okay, we will. Okay, we will start again. For the second part of the conference. Tout le monde voilà, regagne son siège. Merci beaucoup.
So if you could all take up your seats again, thank you. So for this second part, we'll be dealing more with the microbiome, new approaches to non-communicable diseases. We have three professors with us, Maria Yazdanbaksh, almost, Susan Lynch, and Michael Wagner. I'll start with Maria Yazdanbaksh. So within the Netherlands, you're head of the parasitology department at the University of Leiden, working on interactions between parasites and their human hosts. What's the role of the helminths, intestinal worms, for the building up of immunity, and what about the presence of these intestinal worms in early children? To what extent it can prevent inflammatory diseases and allergic reactions?
Well, first of all, thanks very much for the invitation. It's really an interesting gathering and meeting. And indeed, I'll be talking about parasites. That's what me and my team are very interested in Leiden University Center of Infectious Diseases, and also their interaction with the immune system. How do I go to the next slide? So, parasites. Not many people know about parasites, but a lot of people in the world are infected by parasites. Quarter of the world population is affected by parasites, and these are organisms in very different shapes and forms that can live with us for a long time. Some of these parasites are also interacting with our microbiome and changing its diversity. And for studying parasites, about 1936, there is a paper from the Netherlands where 70% of the school children were infected with parasitic helminths, parasitic worms. So that's not so long ago that in Europe we also had these parasitic infections. But we can find them no longer here. We've lost them. And if we want to study parasites, we do that by going to low middle-income countries. And those are areas which really showed us a huge difference in biodiversity. So in these countries where we study parasitic infections, you can see in urban settings, urban centers that you would also, for example, the lower left side, you see Jakarta in Indonesia, a mega city, which is a metropolis where you would not see that much green, and you go a few hours further and you have a very rural area, the lower left side, where there is great, you don't need to measure any diversity. You can, by diversity, you can see is immediately very distinct between these two places. The same in African countries, in Uganda, Kampala, capital city, which has less biodiversity than when you go 2 hours further and you see fishing villages, rural areas where the impact of the biodiversity in the environment is enormous. And in these particularly rural areas where there are a lot of parasitic infections. And, it's really, really very important for our work in any case, where we are trying to understand the relation between parasites and the immune system, and we come across biodiverse areas, and we can really look at the interaction of biodiverse areas, their impact on the immune system. But we can do this really by having very good relationships, very good collaborations. We're very lucky to work with best of minds coming from the global south who are are interested in biodiversity and the interaction with the immune system. So we have lots of connection, lots of networks, which allows us to look at the impact of biodiversity, which seems to be very good for health, as we heard in the earlier session. How does it work? How does it impact the immune system? So it's not so much what's in the biodiverse area is the good factor per se, but also you can turn it around and say, what does the immune system look like that is, as a result of interaction with a more biodiverse region that is really stopping the immune system overreacting to various pollens, allergens, but also maybe your own body. You don't turn onto your body. So this is, what I would like to show. And it's as a result of these collaborations that we were able to do this work. And the immune system, very put simply, because not everybody here is an immunologist, is basically you get some of these infections come in, and in the environment where there's lots of microorganisms and parasites, they're challenging you, the immune system gets activated, it really starts getting prepared to attack some of these pathogens, and by doing that, you can control the microorganisms and parasites, and then when it stops, the immune system is still going on, is producing stuff to, kill the pathogens around. And what it can do, once the pathogen is gone, can also cause damage to the immune system. So the immune system has got a regulatory arm. So we have the attack arm, but we also have a regulatory arm. And the regulatory arm, these cells, become activated and stop the inflammation. So then everything comes back to the homeostasis. And this, if you're in a very diverse environment where you're exposed to microorganisms and parasites, you can imagine that this dynamic interaction is going on so much more than if you are in Jakarta or in Brussels, where there's less interaction with some of these microorganisms and parasites. So when we look at the immune system together with our international team, look at the immune system in less biodiverse areas and in more biodiverse areas, we see indeed that there is much stronger network of the immune system interconnected cells in more biodiverse areas because of this continuous back and forth of activation regulation. And in less biodiverse areas, that's not the case. And if we use some fantastic methodologies that we have here in high-income countries, collaborating with low-income countries, we can look at the immune system, for example, of Europeans and Africans. And if you don't have to be an immunologist, all you have to do is look at the differences between the upper row, which is European, and the lower row. And what you can see, I think just visually, look at it as a painting, you already see that chunks of areas in the immune system are missing in the Europeans. ILCs is one panel of cells that are lacking largely in Europeans, but they're highly populated in people who live in Africa, in biodiverse areas. And that goes across many of these cells that we see. So basically the message is the immune system in people who live in a more biodiverse area versus less can be greatly different. And if you look at European and African, you say, oh, it's genetics, of course. But if you go within a country, you can also distinguish rural from urban areas. This visual image of how the immune system separates from Europeans, from urban Senegalese versus rural Senegalese, you see that urban and rural Senegalese are also showing big differences in their immune system. So it cannot be just genetics. So we heard how important it was that, the immune system, that the allergy, for example, you respond to allergy in areas on farms where there's more biodiversity. You don't seem to develop as strongly asthma and allergic diseases. Now, I heard about that a lot. Well, in rural areas of low middle-income countries, there is hardly any asthma. So, we did studies in Indonesia, in African countries, and every time in rural areas, allergy, asthma. So in biodiverse areas, in those areas, there's also very little allergy. But I want to talk about another respiratory problem. I'd like to talk about COVID. So you know that COVID-19, when it hit, there was a huge, and rightly so, worry that it will affect African continent disproportionately. It's going to be a disaster. Well, what happened in the end? The deaths due to COVID-19, this is shown here. So in France, United States, and Europe, you see many more deaths compared to Africa, to Gabon and Senegal. If you get the data, you see that the number of deaths in Africa were not as huge as in Europe or in the US. So I'm not saying COVID-19 is not a problem. It was a problem because of economic problems, because of other diseases were not being dealt with, but causes of COVID-19 deaths were not there. We heard from colleagues, you know, it was not filling all the hospitals. And there was a commentary from a colleague, Mustafa Mbo, who really called me up and said, Maria, the hygiene hypothesis is working because, you know, we see a lot of infection, but we don't see people dying because of COVID. We should really look into that. And indeed, we started, and, you know, why is it that there is, you know, much less deaths in Africa? People say underreporting, right? Yeah, there's not a good health infrastructure, so there's not people die, but they're not being reported. And a study recently in Zambia, they did a post-mortem of 70 individuals, so people who had died, and they found that 15 to 20% were positive for SARS-CoV-2. But then the question is, are these people dying with COVID because everybody was infected, or are they dying because of COVID? So that's a very important, you know, question. And I know Africa, you know, it has a poor health system, but they really do a great job. We even can learn from how, with little resources, they can, you know, deal with reporting of public health events, Ebola, Lassa fever, pneumonias. They are reported. So, and also, as I said, you know, colleagues from hospitals, we work closely with colleagues in hospitals. They closed the wards that were opened for dealing with COVID. So what about age? Age is an important factor that, you know, we know that older people are dying from COVID. Well, in Africa, it's a youthful population. If you look at the age structure in Europe and the US, it is really, we have a lot more elderly compared to Africa. Well, if you do a calculation of the standardized age mortality ratio, you will find that the difference in the age groups will predict that you have 4 times less deaths in Africa. But it's not. It is 20 to 40 times less. So age is important, but is not the only factor that is explaining this large diversity in discrepancy in death. So genetics. Genetics might be very important, but it was very quickly reported, as you know, in the media, and there are many papers, but this one, that, you know, Black Americans are being affected by COVID at a much higher rate. Well, you know, if, for example, looking at some regions in some regions of the US, Chicago, for example, 32% of its inhabitants are Black, and it was 67% of them that were dying. So disproportionately higher number of Black Africans were dying from COVID. So genetics cannot be the factor that is explaining the low deaths on the African continent. So then we go to the immune system. Sorry, So the immune system, which is something we've been studying, and when I showed you, there are really great differences in the immune system of people who live in these diverse and biodiverse environments. And let's have a look and see, what happens. So we did a study of age and sex-matched individuals. So that's very important, age and sex-matched, and identical scoring of all the symptoms and identical lab procedures. And you look at the immune profile in a multi-dimensional space. So please just look at this here, uninfected European, their profile. Uninfected Senegalese, the profile is different. I showed you Africa and Europe, they have different profiles, slightly different. When it comes to the ones who become infected, the infected Europeans react hugely differently from uninfected Europeans. Their immune signature is very different. They react to the virus. When you look at this in Senegal, they're just very similar to uninfected Senegalese. So these are infected Senegalese with mild symptoms in Europe also. These are not the severe, and they're just as though they haven't seen the virus. And what happens if you look at the inflammation? So you can look at biological markers, and you see that in infected Senegalese, much less inflammation. But if you look at infected Europe, much more inflammation. So people in Europe are reacting very strongly to the SARS-CoV-2, but in terms of inflammation, but not in infected Senegalese. And there is immunological recognition. It's not that they don't recognize it. They do recognize it. They have T and B cell responses. But the key is there is not this overt inflammation. And we need to understand that. It's really important because, you know, the biodiversity is leading to an immune profile that is just not reacting crazily to the SARS-CoV-2, but it is controlling it to some level. So this is the situation. You have a highly networked immune system in rural areas, and you have less network immune system in less biodiverse areas because it hasn't learned to see a lot of microorganisms and parasites. The COVID virus comes, and there is a different reaction, a much more exaggerated inflammatory response in the urban areas compared to the rural areas. So with that, I would just like to say a few words. And, you know, I think from what we heard this morning and from a lot of studies that we've conducted, I think many people have conducted, that, you know, it's clear that the changes in our environment and lifestyle has dramatic impact on our exposure to microorganisms and parasites. The biodiversity index is changing, and so is our challenge to our immune system. And the exposure to these biodiverse areas is really changing our immune system, which becomes more interconnected, cross-reactive, and can fend off pathogens. When you perturb by allergen or by SARS-CoV-2, most of this interconnected immune system doesn't react out of balance. It's well balanced because it's been exposed to so much microorganisms and parasites. So what do we need? What do we need to do is really, I mean, I think like other colleagues, we need to basically understand how it works. It would really be important. I understand there has to be interventions, but please, when we do interventions, we should also try and understand the mechanisms, because we need to understand it's the immune system that is really being trained in a different way in a more biodiverse environment, and we don't know how yet, but I think we are making progress. We have a lot of possibilities to look at that. So it's really important to look at that, identify the pathways that we can mimic to prevent diseases. Maybe we know which immune system do I want so that I do not overreact to an allergen or I do not overreact to a virus. And therefore, we here in the European Parliament, of course, it would be really good to have long-term plans that really allows scientists, not only from Global North, but let's partner with Global South, because that's where the most biodiverse environments are. Huge numbers. We don't have 100 people or 200 people. No, there are millions living in areas where you can learn so much more. So partnership with them to work together to understand, you know, from experiments of nature, as well as, you know, interventional trials, what we need to really facilitate a better health through biodiversity. And with that, I also want to thank the funders. And, you know, European Union is a real great funder of some of these activities that I showed. ERC has been a great source of funding, and I hope, you know, the European Parliament will support the existence of ERC, which is doing a great job. But we also have other EDCTPs and other European funding agency, and of course colleagues who have been helping, and thank you for your attention.
Merci beaucoup. Thank you very much. Now, Susan Lynch, You run the Benioff Center for Microbiome Medicine at the University of California in the US. You're working on the role of the microbiome in the origin of chronic inflammatory diseases such as childhood asthma or inflammatory bowel diseases. Why is the study of the microbiome crucial to preventing and treating these diseases?
Thank you so much. Can I get the pointer? Yes. Great, thank you. Perfect. Thank you so much for the introduction and the opportunity to be here with you today to share some of the work that we've been doing to really try to understand how and when microbes influence allergic disease development so that we can develop interventions to prevent disease development. So really our entry into this field came from observations by colleagues of ours in the United States in which they showed that in urban areas, children that were exposed to dogs and to a lesser extent cats in very early life were protected from allergy and asthma development in childhood. So we hypothesized as microbiologists that this was because dogs and cats introduce microbial diversity into the environment, and homes with no pets present were really the veritable microbial desert. And that's exactly what we showed, as I'm showing here in the center. You can see that in homes with no pets present, the house dust microbial diversity is significantly lower than that in which dogs are present or cats are present. We brought these studies further, much as you'd heard before from Dr. von Muchus, to ask how this dust could actually be protective of these in these environments. And what we showed is that if we orally supplemented mice with house dust from homes with dogs present or no pets present, we saw a profoundly different response in the airways of these animals after they were challenged with an allergen into their airways. And I'm showing this in these panels on the top where you can see, again, those inflammatory cytokines are really muted in the animals whose microbiome was supplemented with house dust from dog-owning homes. They had much less mucin secretion. And as you can see from the pictures below, the control animals, they're the air spaces in their airways. They look pristine. Those that we'd induced allergic inflammation in their airways, those air spaces are now occluded with mucin, and there's inflammatory cells around them. But those that had the dog-associated house dust, you can see that their airways looked like they'd never seen the allergen. They were completely protected against allergic inflammation. This coincided with a shift in the gut microbiome of these animals, and so that led us to begin to think about when in early development does the gut microbiome start to shape airway responses to insults like allergens and viral respiratory infection. And what we've known over the past kind of 15 years or so of microbiome research is that the microbiome develops in very early life and evolves constantly at a functional level throughout lifespan. But over the first 3 years of life is a critical period in which the microbiome develops in the gut. This is when we accumulate microbes and their functions into the gut microbiome. And it's also a critical period of immune training. This is when we train our immune responses to respond to microbes. And so this suggested to us that and also the other point being, as you heard from the speakers earlier in the earlier panel, many of the environmental exposures that relate to susceptibility to allergy and asthma development are really centered in the very early life period. Things like antimicrobial administration and formula feeding increase risk of allergies and asthma, while exposure to livestock and pets in the home decrease the risk. And that suggested to us that really to understand a disease that's diagnosed in childhood, we really need to pull the timeline back to very early infancy and understand early microbiome development in the context of disease susceptibility. And so the first study we performed to do this was a longitudinal study of infants where their stool samples were collected at birth, 1, 3, 6, 9, and 12 months of age. We profiled the bacteria that were found in their gut microbiome, and what was really staggering to us was that in the meconium of these babies, this is the first bowel movement of a newborn baby, their bacterial microbiome is significantly different if they're at high risk for allergy versus low risk. They're deemed high-risk babies if they have at least one parent who already has allergies or asthma. But this difference at birth does not stop there. it continues throughout the first year of life and seems to be compounded with advancing age. We see that babies who are at high risk for allergy development fail to diversify their gut microbiome at the same rate as those who are at low risk. This means they're failing to accumulate microbial functions that modulate immune development in this critical period of life. We had another interesting observation in this study was that those children who were at high risk for allergic disease development lacked exposure to pets in their home, consistent with the epidemiological observations that had been made some years before by our colleagues. In a second study, we really advanced our further knowledge in the field by looking now cross-sectionally and at a much larger number of children, 130 one-month-old infants, in which we profiled both their bacteria and their fungi in their fecal samples. This was a longitudinal study, so we knew whether they developed allergic sensitization or atopy at 2 years of age and asthma at 4 years of age. And we took a hands-off approach to the analysis of this study. We asked an algorithm whether there were different microbiological states that were evident in these one-month-old babies. And we found that there were 3. We called them neonatal gut microbiome 1, 2, and 3. We next asked whether any of these microbiomes related to risk of disease development, and found that babies at 1 month of age who had this neonatal gut microbiome 3 composition were at significantly high risk of developing atopy at 2 years of age and asthma at 4 years of age compared to either of the other 2 groups of babies. So what was different about this group of babies? Well, again, in a completely independent cohort, they lacked exposure to dogs and pets that diversify the house dust microbiome in their home. These microbiomes that increase the risk of allergic disease and asthma development years later in childhood were also reflected the environment of these children. They lacked a whole range of bacteria and bacterial functional genes that, again, modulate immune function. They were enriched for a number of fungi that we know are allergenic fungi, things like Candida and Rhodotorula. And they were metabolically distinct. We looked at the compounds produced by the microbes in these microbiomes and showed that they were vastly different if you were a baby in the high-risk group versus the lower-risk groups. Specifically, we saw that the high-risk groups were increased in a range of inflammatory sugars like fructose and monohydroxy fatty acids. And again, as has been shown earlier on, they were depleted of these anti-inflammatory lipids like polyunsaturated fatty acids and a number of steroid metabolites that we know dampen inflammation. We went on to show in this study that if we took these microbial products in the feces and we added them to immune cells, human immune cells, the high-risk neonatal gut microbiome could actually provoke the kind of quintessential immune dysfunction that is associated with allergic asthma. As you heard by the previous speaker, that is lower regulatory T cells, so a failure to mount T cells that can reduce inflammation, and higher numbers of these inflammatory Th2 cells that provoke allergic inflammation. So this suggested to us that from the earliest stages of postnatal life, these babies have a pathogenic inflammatory microbiome in their gut that's driving allergic inflammation from the earliest stages of life. And we don't diagnose this disease until years later. So we think that there's a snowball effect over those initial years that leads to the diagnosis later in life. And I should say, in work that we have just recently completed and has been accepted for publication, we see a very similar trend with childhood obesity and overweight phenotypes. Very early life microbiomes at 1 month of age predict disease development and shape physiological features consistent with obesity. But this early life microbiome dysfunction needs to come from somewhere. And we know that the majority of microbes found in very early life microbiomes in the skin, in the airways, in the gut, are sourced from the mother. What's also compelling to us is that the environmental and the other exposures that influence the risk of disease development are centered also in the prenatal period and also are strong selective pressures on microbiomes. So this suggested to us that perhaps we need to even look earlier in human developmental time and begin to look during pregnancy and the microbiomes of the mother to ask how they may influence susceptibility to disease development. So in a large study of 172 mother-infant paired samples, in which we had vaginal samples from the mother and stool samples from the infants collected when the babies were 1 to 2 months of age, we examined the bacterial microbiota in these paired samples. We had a whole range of maternal factors, including the maternal health status, her exposures, her stress levels, and farm exposures in this study. We also had several metrics for the child, including total serum IgE. It's an early marker of allergic disease development. For a subset of these samples, we went even deeper to look at microbial functional genes encoded by the microbiomes in the vaginal swabs and paired stool samples of a subset of children and mothers in the study. Again, the first thing we looked at is, can we divide these individuals microbiologically into distinct groups? And this has been described before in the vaginal microbiota. And here we can see in the last weeks of the prenatal period, 36 to 39 weeks, we see 4 distinct vaginal microbiota states that are evident in pregnant individuals. We name these vaginal clusters 1, 2, 3, and 4. What was exciting for us is that these vaginal microbiota states relate to the mother's health status, to her depression and stress levels, to farming exposures, including pig exposure and lack of farming exposure, and to SIBSHIP. And we also had the interesting observation that one of these vaginal microbiota states was predominantly found in women who consumed non-animal milk. And that group had the highest proportion of infants who were cow's milk intolerant or allergic. So really suggesting that diet of the mother shapes her microbiome and influences offspring allergic inflammation phenotypes. We next asked whether the types of microbes that are transmitted from the mother's vaginal tract to the infant gut microbiome actually shape risk of disease development. And here is just simply every column in this plot is a mother-infant pair. Every red square is a bacterium that's found to be shared between the mother's vaginal microbiome and her infant gut microbiome. You can see that there's quite a number of them. We next asked if the pattern of microbial sharing between mother and child relates to a number of features, and found a resounding yes. So the type of vaginal microbiota present in the prenatal period just before birth relates to the types of microbes that are transmitted to the baby and that survive in the gut microbiome up to 1 to 2 months of age. So this suggests to us that environmental exposures, diet, and maternal health shapes transmission or microbial inheritance at birth and in the early stages of postnatal life. Critically, we found that maternal health status, and specifically whether the mother had eczema or asthma, related to these transmission patterns, and also her pre-pregnancy BMI related to the types of microbes that are transmitted from mother to child. And crucially, we saw again that this transmission pattern related to whether the baby went on to develop early life markers of allergic inflammation. So this suggested to us that this is the first evidence for microbial transmission of disease, contributing to transmission of disease from mother to child. And then finally, we examined what were the specific microbial traits that related to whether the baby went on to develop allergic inflammation or not in very early life. Crucially, what we saw was that babies who did not develop allergic inflammation inherited microbes with the capacity to metabolize histamine. I'm sure many of you know when you have allergies, the first thing you take is an antihistamine. These babies inherit microbial capacity to degrade histamine and reduce inflammation associated with histamine. They also inherit microbial capacity to metabolize stress hormones, suggesting that they have their own system that is microbially transmitted to regulate stress. And lastly, those who didn't develop allergies had inherited the capacity to produce a number of fatty acids, several of which we know to be anti-inflammatory. On the other side of the coin, those who had mothers who had lack of exposure to farms, they inherited microbes with the capacity to produce simple sugars that we know to be inflammatory. Glutamate, which we know now activates the T cells that can provoke allergic inflammation. And prepillin peptidase. These make microbial products that provoke inflammation. So this really suggests to us, though it's a relatively small study, that there is evidence for microbial transmission from mother to child and the transmission of pathogenic microbes from mother to child in those mothers who do not have exposure to biodiversity and farming environments in the prenatal period. And this is essentially our current proposed mechanism of disease heritability. The biodiversity and prenatal exposures of the mother shape her health, her microbiome, and her immune status. And there's heritable features of all of those facets. There's heritable microbes, there are heritable epigenetic modifications and immune functions that are transmitted from mother to child. And then these govern the early life microbiome development and immune development of the offspring, which is continuously shaped by the postnatal exposures encountered in the living environment. And so, lastly, I just want to acknowledge the biodiversity of investigators that it takes to mount these kinds of studies and execute these kinds of studies. This is a group of epidemiologists, immunologists, microbiologists, clinicians, and statisticians. And it really is in recognition of the group effort that it takes and the interdisciplinary nature that's necessary to perform these kinds of studies. Thank you for your attention.
Merci beaucoup. Enfin, Michael Wagner.
Thank you very much, Michael Wagner. You're a microbiologist at the University of Vienna in Austria, and you lead an extensive research program, How the Microbiome Directs Global Health. So there we are, we're taking on a global dimension. So what's the aim of the program on the microbiome and planetary health? And why did you choose that particular title? And why do you want to combine medicine and ecology?
Thank you. Last speaker, so I'm excited to be here today and look forward to discuss a little bit with you a forward-looking perspective on microbiomes. We've heard a lot, about the connections of environmental microbiomes, exposure to cows, to raw milk, and consequences for allergies, for example. But if you think about it, there are also many difficulties associated with this, links. We have benefited a lot from hygiene in the past, right? If you look at the development of life expectancy. So the answer is not simple. You cannot simply say, hey, to a pregnant woman, drink raw milk. There is no risk associated with this. There is a risk associated with it. So we need smarter solutions. You cannot say in a Chinese city, all of them should drink raw milk. How would you organize this? There are these health issues, pathogenic bacteria in the milk. So we understand, and this is the value of all these studies, the price we pay for hygiene, which we need. So where do we find the right balance, and can we find smarter ways to manipulate our microbiomes to protect our health? But for that purpose, we need to understand them. And we have formed a large consortium in Austria, which is entitled Microbiomes Drive Planetary Health. It's a cluster of excellence. It's funded for the first 5 years with 35 million euro. I have the pleasure to be the director of this cluster. And I would like to share with you a little bit our vision, what we want to achieve, and then show you one example, how we study microbiomes and how we think we can make a contribution to human and planetary health. First of all, just want to remind us, this is a planet of microbes, right? They were here long before we were here, billions of years ago. They will be here when we are gone. And if you would put them all in a way, they determine their biomass, right? They are as heavy as all animals and humans together. They are tiny. They are 1,000 millimeter large. But if you take them all together, they are as heavy as all animals and all humans, which already tells you how important their physiology is for the element cycles on this planet. And there's a huge diversity out there. In 1 gram of soil, so if you go home tonight and you look at your flower pot, in 1 gram of soil you find 100,000 bacterial species. 100,000 species in 1 gram of soil. A few handful of soil contain more bacteria than humans on this planet. So if we want to understand these hyper-complex communities, we need high-tech, right? This is not a simple task. These are enormously complex communities in the environment, in our body, and they are all connected with each other. So this is a whole field of research, and we tackle a very complex challenge here. We have heard in the first session about this concept of planetary health, which is the health of the human population, here the human in the center, and all the systems where this human population is embedded in. But then this is a figure from a Lancet journal, and if you look closely, it all makes sense, right? Humans are affected by climate, by urban environments, by animals, by plants. But what is missing? If you look closely, there are no microbes. So it's shocking, right? Even today, in such figures, often microbes are totally overlooked. So this figure actually has to look like this, right? Microbes are essential. They are essential for our health, for the health of the planet, and they connect all the players and determine in large part how these systems respond to its many stressors, like global warming, growth of the human population, and so on. So we need to understand these microbes much better. And that's what we want to do with our Microplanet Consortium. We are 30 principal investigators from 8 leading research institutions in Austria, and we are all focusing on understanding microbiomes in the environment and in humans. And we tackle 3 big questions, which are really the big unknowns in microbiome research. This is microbiome interactions. We can now, with molecular tools, determine which microbes are in such complex systems. We can make sort of a stamp collection, right? But how do these microbes interact with each other? And with their hosts? That's much more difficult to extract. Then, how do these microbiomes respond to perturbations, right? Global warming or pharmaceuticals, or you can name them, these perturbations. And we need this knowledge, and we need to extract general principles from different microbiomes in order to design targeted manipulation strategies. Because the goal must be, right, we want to restore a healthy microbiome, even if you live in a city. We want to, have better microbiomes in wastewater treatment plants, in agricultural soils, you name it. So we don't only want to describe the problem, we want to solve the problem. For that purpose, we need to understand this complex system. This is, by the way, a picture I took during my PhD thesis of a wastewater microbiome, which shows you how these microbes, look at the bar, they are very small, live next to each other and interact with each other. Just 1 second, I want to show you what is different in our consortium compared to other consortia. We think this separation of environmental microbiome research and medical microbiome research is very harmful. We have to tear down these walls. We know microbiome researchers, if you're in this field, they work in silos. They either work in the medical field or in the environmental field. But basically, these are all microbiomes, and they are all connected, right? We get the microbes from the cows and so on. So we designed this research program in a way that researchers, which you see here on the left side with their names, which have expertise in the environmental field, in the health field, and in the method field, all work together. Every project has researchers from each of these domains, red, green, and blue. And I do think this will bring quite some innovation in this field. So how do we study these microbiomes? And this seems to be technical now, but please stay with me for a minute, because what everyone is doing is sequencing those microbiomes, determining their genes, right, their genomes. This is now sort of state of the art. Every lab is doing it, and it's necessary. I'm not criticizing it, but often people think this is enough. So you get catalogs of genes. But for many of those genes, you have no clue what they are encoding. And for those genes where you have a clue, you think you have a clue, you often nevertheless don't have a clue. This is one more science slides I will show you in 1 minute. From these genomes, the field, reconstructed the tree of life. So have a look at this tree of life. I would love to have a pointer now. But you see in the upper part of this tree of life, which shows the actual relationships of all organisms on this planet, right? You see the bacteria, this big cloud of branches, right? These are all bacteria. And then on the bottom, there are also other unicellular organisms, the archaea. And then you see one branch, the green one on the lower top right? And these are the plants, the animals, and the humans. So when you think about biodiversity, right, we usually think about plants, animals, and humans, but it's just one small branch in the tree of life. And what is actually biodiversity? Look at the poster here. Biodiversity is microbes, right? Because they had much more time to evolve this enormous diversity. We just don't see it because they are so small. But when you talk biodiversity, you talk microbes. You don't talk animals, and you don't talk plants, you talk microbes. However, when we get these genes, and these two slides were flipped, when you get these genes from the genomes, and you think you understand what the organisms are doing, you are very often wrong. Why is this? I spent some time at Caltech. There's Frances Arnold. You might have heard of her because she got the Nobel Prize. She does evolution of what her work is. She evolves proteins in the lab. And what she does here, and it sounds complicated, but the message is very simple. She took a protein, an enzyme, cytochrome P450 from a bacterium, and changed only 2% of its sequence, of the amino acid sequence. And this totally changed the function of this protein. She has many of these stories. So what does this tell you? If you have genes which are very similar to each other, most microbiologists say, oh, these organisms can do the same thing because they have highly similar genes. In reality, often the organisms do different things. So what does this tell us? If you just sequence genomes, you have to do it to get an hypothesis, but this is not enough. You need more direct ways to analyze the function of microorganisms. And this is something we are doing in our consortium a lot. And no look at the details, but we developed, for example, this is together with the Boston Photonics Group, new instruments where we can directly measure what microbes eat in a complex community. So we observe these tiny organisms, 1 micrometer, 1 millimeter divided by 1,000 large. We observe them while they eat and learn what they consume and what they produce with these instruments. So data look like this. On the right, the gray picture is just a normal microscopic picture. You see the gut microbiome, you see cells, right, sitting next to each other. On the very left side, the blue picture is a molecular probe which identifies the microbe. It says your name is, for example, E. coli. And the yellow picture shows you the actual activity of the microbes. So this is not a fluorescent picture. This is an activity picture. And you record this activity in real time, so in video rate. So then you can add different substances to your sample, and you can, in very quick way, see how the activity of defined microbes changes, right? So you can measure how the microbes perform in their environment under different conditions. And I show you one example how important it is to do this. Oh, there is a font issue here. But so we study the drug microbiome host network. We heard a lot about factors which influence our microbiomes, right? Exposure to cows, exposure to dogs. But also if you take drugs, and I'm not talking antibiotics now, there everyone knows it. I'm talking human-targeted drugs. So if you take a drug, then it often changes your microbiome. Although it's targeting a structure in your body, not the microbes. And often also the microbes determine whether the drug works in you or not. 40% of all patients over all drugs do not benefit from a drug treatment. And whether a patient benefits from a drug treatment or not is highly individual often. And one factor which contributes to that is the microbiome composition. So it's important to study this interaction of drugs, microbiomes, and humans. Microbes can convert drugs, they can decay drugs, and drugs change microbiome composition and function. One example, I just picked one example of a recent research study we did. This is entacapone. This is a drug you use for treatment of Parkinson's disease. And it's widely used. This is a complex figure which you see here, but just look at the colors. On the left side, you see the composition of the human gut microbiome at the start of the experiment. In the middle, We took a fecal sample and we incubated it with a growth medium. You see in the middle, after 1 day, 24 hours of incubation, and you see all the colors get warmer, right? Almost everything is red. That means, and that was what we wanted to do, all the microbes in this fecal sample were growing, right? They became more abundant. That's why the color becomes more red. And on the right side, you see the same incubation, just 24 hours, human fecal sample, same medium, but with entacapone. And a therapeutic concentration of entacapone. So we use the concentration patients find in their large intestine. And you can see how dramatically this entacapone affects the composition of the gut microbiome. And not surprisingly, entacapone has many side effects, gastrointestinal side effects, because you change the composition of your gut microbiome in a dramatic way. And many drugs do it. This is just one example. And when we looked closer, and we have tools to do this, we can visualize the entacapone. And we saw that some microbes accumulate in the gut this drug, right? They pump themselves full of this drug. So they remove it. The human is supposed to take up the drug, but the bacteria take it up. So why are they doing it, and what is causing this effect on the human gut microbiome? And we looked at the structure, and we came up of the drug with the idea that maybe entacapone is complexing iron, so causing an iron deficiency in the large intestine. And we tested this idea. And again, this is a bit a complex figure, but the message is very easy. On the top row, you see the 25 most affected taxa in this experiment. This is the x-axis. The larger these bubbles are, and the more warm they are, the more inhibited these microbes are with entacapone. And the line in the middle and at the bottom is the same experiment, but after addition, entacapone, the drug, plus iron. And I think you can easily see the bubbles shrink, the colors are getting less warm. So the inhibitory effect of entacapone on these gut microbiome members is gone. You can almost completely rescue this shift in microbiome function and composition by addition of iron. So what I want to show you here, this is not the solution for all drugs, right? This is a specific effect of this drug. But when we understand why drugs and how drugs impact microbiomes in our gut, we can also find solutions. And this will hopefully then ameliorate side effects and help us to make drug treatments more efficient. And so this is the model for the entacapone. On the left side, you see without entacapone, bacteria in the gut have sufficient iron, and you have a normal gut microbiome. If there is entacapone, it binds iron. We have directly shown this. And then only microbes can thrive which have special molecules to take up iron, siderophores. Unfortunately, those microbes often also carry virulence factors and antibiotic resistance genes. So we made a virulence factor index and saw that entacapone selects for potentially pathogenic microbes. So you take a drug against Parkinson and your gut community changes a lot towards pathobionts, and you're more prone to gastrointestinal side effects. And we hope from this finding that we can now, and we will do this together with clinicians, design sort of approaches to ameliorate these side effects. Last slide. Many factors influence our microbiomes: the diet, pharmaceuticals, where we live, to what we are exposed, how, whether we were breastfed or not, and so on. I think we have quite some knowledge about the changes which are induced by these factors, but we have little knowledge how to reverse them. And this is, I think, a field which requires a lot of funding, a lot of research activity, and where the next decade, I think, should, at least microbiologists should focus on how can we revert these factors and help us to live a more healthy life. And the last slide here is an acknowledgement. I was happy enough to be the founding dean of a new faculty at the University of Vienna, which is completely devoted to microbiome research. It's called Center for Microbiology and Environmental System Science. You see our new building here. We are about 250 microbiologists. And the ERC has been mentioned before, and being here in Brussels, I need to say this, we have 10 ERC grants, and we couldn't do our research without the ERC. So often you ask, why is fundamental research important? We should do something which has a direct effect on the society. Without ERC money, many of these fundamental findings, right, which provided the basis for what we discussed here today, would not be available. So please support the ERC also in the future, because it's essential for research in Europe. With this, I would like to thank you for your attention, and I'm happy to take any questions you might have.
Merci beaucoup.
Thank you very much to all three of you for your presentations. I'll now take some questions, if there are any in the room.
Oui.
Yes.
Vous venez de parler de spoke about using iron to help some of the bacteria populations that were diminished through that drug. I was wondering if you could expand upon that, as well as perhaps talk about a prebiotic fiber in relation, in the same sort of idea, iron, prebiotic fiber, and the importance of that.
Should we collect questions first, or should we
Yes.
answer?
Other questions? Alors, du coup, moi j'en ai une.
Well, I have one from Maria.
In English. It's you finish with a cooperation with African medicine, and it's very instructive to see that African medicine have more to learn us to struggle among disease. You name Ebola, and the example of the COVID is almost clear, because we are in Europe and in Occident, all our hospital and difficulties, and I have the sentiment that we never heard what happens in the other space of the world. So see if you can talk a bit more about this. Je vous laisse.
Yes, another question?
Oui, bonjour, je vais m'exprimer en...
I'll speak French. Thank you for your presentations. Just a thought for Michel Rivasi, who I knew well and who was always very interested in considering new cutting-edge research questions. So thank you to her for having started the ball rolling here. I have a specific question on the GMOs. We have a proposal for European regulation which would cover the change of 20 nucleotides that would have no impact on these plants, and that these plants should be considered as non-modified. But what you've shown is that by changing a very small part of the genome, that we can have extremely significant consequences and unexpected consequences. So what would your reaction be as a scientist to a proposal that's so approximate, as we've seen come from the Commission, and which will have to be voted on at the European Parliament level? So I'd just like to get your reaction to that.
Yes, I have another question. We were talking about needing for children at a very young age to get exposed to bacteria, and the signification is huge. So it would be nice to get all of our children in the cities exposed to the proper bacteria and microbes at a certain amount, maybe once, twice, 5, 100 times a year. We don't know how much this is. But I would like to reverse the idea and ask you if it's imaginable for to bring the microbiome of the farms, etc., into the cities, into the urban environment, and even into the households in order to make it a lot easier than having all of the children go to the farms, maybe bring the farms into the city? And would that possibly work? And how to do it? Thank you.
Okay,
Yeah. May I ask? We'll start with the iron question, and then I make one comment, but the children would probably mostly be here, but one comment I want to make too on that. So with the iron question, the trick is, as always in science, things are a bit more complicated, right? The drug uptake is mostly in the small intestine in this case for entacapone, and if you add iron just as a supplement, the uptake of the drug would be lower. So the answer cannot be just add iron. The answer must be add iron in a way that it's released available in the large intestine, but not in the small intestine, which there are ways to do that. But so this is what we want to test. But this is more complicated than it's first sounded. And your idea of the prebiotics, that's a good idea, pre and probiotics, but we need to be aware that we don't have the same microbiomes, right? So the vision, what would be the vision? In 10 years, you go to a hospital, you get a drug treatment, you do not only get blood work done, but you get also your microbiome determined. And from the microbiome, they might tell you, oh, you might suffer from those side effects. We better give you a personalized probiotic which fits to this drug, right? You're maybe a group of people with this probiotic. Another group of people requires maybe another pre or probiotic. And that's sort of the vision, that we can make drugs more efficient. And not only drugs, it can work for other things too. Drugs more efficient and less side effects, but in a more personalized manner. But there's a lot of research still needed to get to this point. And that also, and this is the last half sentence I say to this is also related to your last question, because bringing bugs to the city and saying, take them up, the risk, the challenge is how to make sure that the kids also have different microbiomes in their noses, in their guts, that they really will permanently colonize it. And they might do this in different efficiencies in different children. I do think this is one of the big challenges, and we need to understand how to get new organisms permanently established in our human body.
Yeah, maybe I'll extend on that point as well. We did think about bringing the farm to the urban environment for many years, but then we had some data that showed exactly as was just mentioned, that the composition of the gut microbiome dictates how many organisms from the environment are actually taken up and successfully colonized in an infant gut. And the babies at 1 month of age who had the high-risk kind of inflammatory microbiome, they don't become colonized by the microbes in their environment. We think that the microbes are provoking an inflammatory response specifically to exclude environmental microbes from successfully colonizing the niche. So the approach that we've taken is to, from first day of birth, supplement those babies with a group of organisms that encode all of the functional traits that are missing in their microbiome over the first year of life, and to sustain that supplementation for the first year to kind of force a tolerogenic immune response that then allows for colonization by environmental microbes into the system to train immunity and prevent disease development. And that's a trial that's going on at the moment in the United States.
Because I know it's a very interesting idea, and I know that I went to some exhibition at the Technical University of Delft in the Netherlands, and they're actually putting bacteria into bricks to make walls in a house. So once you definitely different people have different susceptibility of picking up the microbes, but you take people, I mean, the same people go to the forests. I mean, in answer to your question, it's an interesting one, and I know there are architects that are thinking about interacting with people to, once the good stuff is identified, to actually build it into bricks. That's all I wanted to add.
I want to address the prebiotic question as well, because I think it's really important. As was mentioned, we really have to consider the diversity of microbial functional capacity that's in the gut microbiome, because that dictates how you respond to anything you ingest, including complex polysaccharides and fibers. And so I think the approach that may be more fruitful is one in which you provide those fibers, but you also provide the cellular microbial machinery to convert those fibers into those anti-inflammatory products that kind of serve as energy sources for the gut and reduce inflammation. So it's not a single approach. It's really, again, thinking about interactions and complexity and the biodiversity necessary to promote the metabolic productivity that drives health in the human system from early life and throughout life course.
Yeah, interesting that how you put it. It's that we can learn a lot from other countries, from the global south also. I want to say one thing, the immune system, we all go to universities here or schools and we study the textbooks on the immune system, and what is in there actually is based on what we know from Europe and the USA. And by going and examining the immune system in other parts of the world, you really get to know the rest of the immune system that we don't know very much about. And you can, take it along into the biodiversity, There's so much more probably microorganisms that we don't know much about by, going to including not only our small tunnel vision, but opening it up globally. I think we'll learn a lot. So that's just learning from the biology, but also practices. Yes, the pandemic came, and of course, with the little resources that some of the countries had, they really know their public health measures, whereas here we, of course, are looking for the most sophisticated, and that's very good, we need them, but sometimes we can learn from simpler things. So for me, most important is, collaboration, working together on this African continent, which is our neighbor, for example, opening up a little bit more to the outside, just to improve our science also, and the ways that we can tackle health. That's what I meant. Thank you.
I'm not, although I was also active during the pandemic and switched a little bit fields and developed detection assays for COVID, but so I'm not a virologist, I say this as a disclaimer in the beginning, but it can be more complex too, before the Africa effect, right? For example, it has been seen that rhinovirus, I think, negatively interferes with COVID. So if you have rhinovirus, for example, waves, then the COVID waves seem to be shifted. So there is viral interference. So in principle, it could also be, right? And this is a question more than a comment. In other countries, and you are also exposed to other, viruses, other infections, this could also interfere in how severe COVID is, right? So it might not be as simple. They might suffer from other infections which have some preventive effect towards COVID, but then the answer is probably not, we should get all infected by these other infections. So it is a complex picture.
Sure, I'm not trying to simplify things, but talking about cross-reactivity, I mean, it's too complex to, give the talk for this audience, sure. I thought. And it is known that in certain areas where there were a lot of exposure to other coronaviruses, that the cross-reactivity has helped to control. So for sure, the diversity is also diversifying your ability to react to certain infections. And whether these infections are very severe or not, that's where we have to decide. If some are not as severe, and it's okay. But yeah, more knowledge is needed. That's basically it. I agree.
Maybe getting back to the plant, there was a plant genetic modification comment. I think none of us is a plant GMO expert. I hope I'm saying nothing wrong. But what I want to say is, as a microbiologist, I'm not so concerned about genetic modification because they modify themselves all the time. If you eat meat, you eat DNA, right? Because in meat there is DNA. If you eat plants, you eat DNA. And microbes even put this eukaryotic DNA into their genomes all the time. They test different DNA pieces. There's nothing we do. They are the masters of genetic modification. And when something is useful, they use it, and if it's not useful, they throw it out of their genome. So you can't control it. You have in your gut GMOs basically all the time. And we should also not forget that if you do conventional breeding, you also genetically modify the organisms, often in a way you do not know what you know, where exactly the modifications are. So from a health perspective, and I'm not so concerned, I'm more concerned from an economic, and we could discuss about this, monopoly and whatever. There are, you know, seed diversity and so on. There are many, there are also negative effects, I do think so, but not so much from a, I think, health perspective. Without going into details, and I'm certainly not an expert on that, but you asked and I wanted to provide some answer and not ignore it.
Merci. On va reprendre quelques questions.
Thank you. We'll take a last round of questions. Massar? Good afternoon, and thank you very much indeed for your presentations and for the clarity of what you've said. For the layman or woman, it's not always easy to follow. I've got a question that's a little bit more targeted towards Madam Lynch. You're currently conducting a study on children that suffer from autism, and I'd like to know whether you've established a link between the microbiome and autism.
Yeah, we do not actually study autism ourselves, but we've been involved and we've certainly read a lot of the literature in the field. I think the biggest evidence for the microbiome in autism comes from a small clinical trial that was performed a number of years ago, in which young children who'd been diagnosed with autism spectrum disorder received a fecal microbial transplant, essentially a wholesale re-establishment of their gut microbiome. And that was based off of evidence that came from Circus Masmanian and others groups showing that there was a perturbation in the gut microbiome of children who had autism spectrum disorder. And gastrointestinal issues are a common feature of children with the condition. But with the fecal microbial transplant trial, what they showed was a significant improvement across a range of different metrics of disease severity. But I think what was most compelling was that they followed these children 3 years out. And 3 years after they'd had their fecal microbial transplant, not only did they still maintain that beneficial effect, but there was even further improvements in several metrics of health in that population. So that, for me, not only established that the gut microbiome is paramount in autism spectrum disorder, but that manipulation of the gut microbiome, you know, is a plausible therapeutic strategy for treating the condition with long-lasting effects in that population. Now, the trial has yet to have been repeated in a much larger population, and there were some aspects of the trial design that were not ideal, but it, for me, was the first proof of principle that manipulation of the gut microbiome in that condition with fecal microbial transplant did have a beneficial effect, a significant beneficial effect for a large majority of the treated patients.
Merci beaucoup. Est-ce qu'il y a...
Thank you. Are there any other questions? Is there anything you'd like to add, perhaps? Otherwise, perhaps we'll move on to the third part and the last part of this round table. And here we intended to have a broader debate on policy options at the level of the Union and member states. He's due to join us in a few minutes. That is to say Nikolai Stefanuta. I hope I've pronounced that correctly, says the speaker. He is a member of the Health Committee of the European Parliament, but in the meantime, I'll give the floor to Panagiotis Chaslaridis. I hope I've not done too badly with the pronunciation. You're a policy advisor at the European Federation of Allergy and Airways Diseases Patients Associations. I leave it to you to introduce yourself. You're responsible for prevention issues with EFA, and perhaps you can tell us a little bit about what you're doing and what you're proposing.
Thank you very much, Chair. Yeah, so the European Federation of Allergy and Airway Diseases Patients Associations, EFA for short, represents over 200 million patients living with allergy, asthma, and chronic obstructive pulmonary disease in Europe. Through its activities, EFA works to address environmental risk factors on health, but also works increasingly so also on biodiversity issues. Indeed, this is an area that represents a growing amount of evidence how the environment affects health, and I actually feel really lucky after this richness of scientific findings that were presented in these first two panels. Asthma, allergies, COPD are diseases with very complex underlying factors. Until a few decades ago, they were supposed to be due to only genetic factors. Now, obviously, there has been a paradigm shift there. Environment has been recognized as more and more of a factor that affects the risk to develop the disease. Now, listening to these presentations, there are several thoughts that came to my non-scientific mind. First of all, from a policy perspective, biodiversity is not only about protecting animals and the environment, like many people might think. It's also about a whole range of issues, the sustainability of food systems, addressing climate change, how we do urban planning, how do we address the built environment, and so on. This holistic view that is needed. Because this is an issue that cuts across many different policy domains and aspects of life. A second point is that while there is a strong and solid body of evidence, it seems that everything is at the stage of fundamental research. As was mentioned earlier, the data that show the benefits of green spaces in urban environments, the positive effect of our contacts with nature in the maturity of our immune system, the importance of consuming raw milk, for example, to prevent asthma and allergy, particularly food allergy, the disparities in the prevalence of and severity of asthma differing between urban and rural areas, the effects of lifestyle. So all of these are aspects, and I think that it is our joint duty to policymakers, scientific experts, but also civil society, to work together to communicate these findings and make sure that messaging arrives in a clear, timely, effective, and actionable way to everyone through our various networks, through events like this. Third, research is advancing, but patients are worried, I must say, because we are vulnerable, and we can see it on many occasions also in our contacts with members. For example, we have this developing discussion on One Health and respiratory health. Respiratory health actually is not very visible in the One Health agenda as the European Union is unfolding it at the moment. Policymakers are not putting, as we think, the right amount of attention on respiratory health, and we are actually a classic example of a community that needs a One Health approach. Final point is that we need indeed to continue supporting groundbreaking research like this, shedding light to the underlying factors of the diseases with the participation of patients as co-drivers of this agenda. One great example that was mentioned in the very beginning of this conference of let's say holistic approach has been the Finnish allergy program from 2008 until 2018. And IFA, together with Dr. Hartel, actually had worked to submit it as a best practice through the European Commission best practice portal. Best practice in the sense of being formally recognized as a best practice by the Commission. It is promoted as such, and also transferability is allowed, is facilitated to other countries as well. My organization represents disease areas the prevalence of which is rapidly increasing. Asthma, especially in childhood, affects up to 10% of children. Pollen allergy affects up to 40% of the population. Food allergy is more and more prevalent, together with emergent allergens, actually, on the food landscape. So to summarize a little bit, first point is that we need cross-policy action to connect the dots among the different policy areas that affect asthma and allergy based on the already existing evidence base. Second, we need a stronger focus on the respiratory aspects of the One Health agenda by the EU. Third, that we need to foster improved awareness on these issues based on synergies between policymakers, the academy, and civil society. And finally, that research needs to continue being supported with the strong involvement of patients. Thanks a lot for your attention.
Merci beaucoup.
Thank you very much. Now, in moving towards our conclusion, I'd like to give the floor to each of our speakers, and if in a couple of sentences you could give us, in your opinion, what the priorities should be in terms of public priorities or the European Union. So I don't know which order to take it in, but I'd like to have a comment from all five of you. I mean, you've got 30 seconds to think about what you would say before I come back to you.
Qui veut commencer?
Who'd like to kick off?
I see Erica is getting ready.
Okay. Thanks.
There are lots of good ideas out there how we could combat or prevent these diseases. What is missing is a link to policymakers, and what is missing is the money that we need to do that. And we need to do this. I agree with patients, that I agree with. And I think there are multiple approaches and there are a lot of innovative ideas out there. And as Susan has stressed, I think to bring the people, also Michael, the people with the different expertises around the table, I think is crucial, but I would include patients and I would include policymakers and I would include, the environmentalists to try to find, to discuss the ideas and then to commonly think of what is the best feasible and the most promising ideas, and then to have the necessary funding to implement this development, because it's a necessary development. That's what I would say.
C'est de la prospective.
Well, this is a bit complicated to forecast because this would involve a necessary cooperation of different disciplines for research, and that's something that is not yet very accepted. I mean, I think the Austrian example is something we should really follow, but we're not there yet everywhere else. And when we have tried for many years to have a One Health approach before it became fashionable and have a multidisciplinary approach, we realized, at least I felt this in France, that we'd come back rather more to the individual disciplines, and institutionally speaking, this mix of disciplines wouldn't be supported, and this mutual understanding, everyone would stick to their own field in so far as possible, but make their expertise available to these common studies. I mean, what we would like perhaps would be the example that Michael Wagner has shown us, that this could be disseminated out to the other European countries and Europe in general in order to facilitate and provide institutional connections so that this work is not just relying on the researchers' goodwill, who are a bit avant-garde in their approach to things and that spontaneously look into things, but that then there's a lack of continuity over time because it's not institutionally supported. A second desideratum, I think this is something Erica agrees with, that would be the problem of financial support for following cohorts. We have a system to organize research by projects and temporary and one-off over 3 years. And it's very difficult to maintain in several countries a common action like PASTURE, but not just PASTURE. And I think that this is something that should really be thought about. We run up against the problem of European regulations as well, in terms of data confidentiality, etc. For children who are then over 18, they need to agree to having participated in the cohort, and it's not the same thing in all countries, so we run up against regulatory problems. So I think somebody needs to oil the machinery there somewhat. I mean, if you want to have research, you need to do this. I mean, if it becomes too complicated from a regulatory point of view, there's nothing left but to stop. I mean, we almost reached a point where we didn't know whether we would be able to continue or whether we'd be forced to stop. So I'd rather not be in that situation. Thank you. Next.
I always try to move the micro and it doesn't move. I think I tried it 10 times. Some things scientists don't learn. So, yeah, first I support these comments. I think interdisciplinary research is really essential. You know, killing these silos, red, green, bringing in many disciplines, translating it to patients and cohorts. That makes a lot of sense and is necessary. And on the other hand, and I would really like to stress this, don't forget about basic science. Where we are here, you know, 20 years ago, people said, microbes, these are pathogenic microbes, we should look at those which make you sick, and all the others, who cares, right? Now we learned these are our best friends, right? The bacteria, we always looked at them from a perspective of these are our enemies, we have to fight them. And now we learn we need to culture them in us, right? We need to grow them, we need to appreciate that most of the microbes are beneficial for the planet and for us. This did not came from applied research, this came from basic science funding. And now we are at the point that we can catalog them, we start to understand these organisms, but we want to sit here in 10 years and report to you, right, that we now can find solutions with this for patients, for the agricultural soils, have less climate gases, greenhouse gases emitting from agricultural soils, and so on. So we need so many solutions, and we will only get there if you let researchers do their work and fund their basic ideas. And this is under a lot of pressure because politicians want answers fast, right? You want to have solutions fast, and you want to see the value for the money. And it often takes 10 years, 20 years. But then we sit here and can report really about breakthroughs. And you need to support us and to support basic research, because this will bring the solutions for the future. So fund the ERC and fight for basic science is my main wish.
I agree with everything that's been said beforehand, and the concept of interdisciplinary research. That's really how we've accelerated our knowledge over the last decade or so. And that requires large programs that are very expensive and require funding. But it also requires that we think about how we educate and how we train our populations in higher levels of education. Education is quite siloed still. There's pulmonologists, there's gastroenterologists, and they don't speak to one another. So I think thinking about how we really cut across those silos, drop those silos, and really think about how we train the next generation of researchers and how we bring populations together, basic scientists as well as clinicians to tackle some of these larger issues. And then the other thing I would say is that I completely agree, long-term funding, it's the same in the US. We go in 5-year rotations, and that is not how we need to do research. We need to understand evolution over lifetimes and intergenerational evolution over multiple lifetimes, as you've seen from what was presented today. These are long-term thinkings and long-term strategies for how we do research, and it's not the 5-year turn and burn approach. And the last thing I would say is funding clinical trials. If we really want to bring products to market that can actually be used in the clinic, that's a 10-year proposition at least to get through a clinical trial. They're very expensive propositions, but they're absolutely necessary if we want to be able to make claims about drugs, microbial or otherwise, that will allow us to halt the tide of chronic inflammatory disease that we're facing.
Well, everything has been said, but I would still like to add, maybe we need to also think, events like this, I think, is really important, right? So we're all talking about, we are scientists, and of course, basic science is important, translational science is important. But if we could have time also to engage with society, but also with funders, I mean, it's really important to have such events that we can talk and as scientists and communicate. I can see that there is interest. And why don't we do that? Because we don't have enough time. And why don't we have enough time? Because I'm afraid, I'm not saying, you know, money, money, money. No, but it is an important part. And let's just think about COVID. COVID vaccine was possible in no time. How is that possible? If that's possible, then if you put your priority right out there, and that's the most important thing, that we want to help the patient, surely there is the possibility. So I think it's also making that, giving people energy to say, yes, you know, we need, just like COVID, how we very quickly managed to develop a vaccine. We have the technologies, we have the knowledge, we will gain more knowledge, but, you know, there's a lot that can be done also. I think that's really also important to add to everything else. And of course, you know, I think we discussed genetics, we thought for a long time was the reason people got allergy and asthma. And now we realize it's the environment, because certain people worked in certain environments. Those environments are disappearing. So please, as I said, we need to look beyond Europe maybe, or Eastern Europe, beyond Europe, to be able to make new discoveries, like we did, saying genetics is not as important as the environmental factors for allergic diseases and asthma that we are discussing today, just as an example.
Maybe just one more thing, and this one more thing is, it happened that most of the development and the investments for medical innovation were through pharmaceutical company, because it was thought about therapies. What we are discussing today is a lot about prevention, and pharmaceutical companies are not interested in this for many reasons. Academic places like medicine, hospitals, are no longer in the position to run these things. As Susan said, this is highly expensive. So the problem is an infrastructure. We need to create an infrastructure, and that has to be the public, the setting, that gives us the infrastructure to work on things where we miss the investments that pharmaceutical companies did for development of drugs. I mean, you realize that developing a new drug costs 50 million or more, right? And the companies, as much as we may criticize them, had the investments to make this possible. they won't do this for prevention. So the public domain needs to think about how to invest in such a field, which eventually will give you the biggest gain, because preventing at the end will give you the biggest gain rather than treatment.
Merci beaucoup.
Thank you very much. I think you've taken the words out of my own mouth, in fact, because I was going to conclude on that note. I think we're picking up, sort of the wage of the battle, which was that of Michel Rivasi, when it comes to issues such as funding research, transversal approaches. These are all things that I think were characteristic of her career, listening to the specialists. She was a specialist herself. In the area of biology, she was trained in there, but she listened to other specialists, patients, citizens. And I think that was what she had to offer. She enabled those to speak out who wouldn't necessarily have been able to do that. But I think we as politicians have to make things sort of accessible, sort of simpler, less complex. That I think is our role. Journalist Marie-Monique Robin is a case in point. There's an educational approach that's required. I think one of the things that Michel Rivasi wanted to do was to stand up to the sort of big pharma and promote a longer-term vision. I think it's certainly true when it comes to research, but I think it's true when it comes to protecting this planet. I think what we need to do is conceive of policies that go beyond the next 5 years. Of course, political mandates, sort of 5 years. And big companies have a lot of financial clout. If there's no democratic counterpower saying that research needs to be oriented in the direction of prevention rather than just coming out with mere recommendations. I think that's what governments are doing. They're making recommendations rather than investing in prevention, with one or two exceptions, as we've seen in Finland. And as we've seen in the case of Africa, where there are no means available, you make do when it comes to ensuring that policies that are put in place are effective. When it comes to the sort of the way pandemics arise, Ebola, for example, I don't know whether in Europe we'd be able to respond to that kind of pandemic. Ebola, it's fairly sort of explosive, and I think the response is found to limit the spread of Ebola. Because I think we had to ask ourselves at some point in time whether it would, sort of transcend the area where it was confined. But I think the responses to the issue were fairly straightforward and simple. So I think we have to sort of learn to live with microbes, biodiversity, animal and plant biodiversity, and respect that biodiversity. So thank you very much for some of you having travelled from afar to be with us. I'd like to thank those of you in the public who have been listening to these various presentations right to the end. And of course, we will be acting at the level of the EU on these issues, I'm sure, in the future. I'd also like to thank our interpreters who've worked with us this afternoon, and let us hope we will have an opportunity of meeting soon on another subject. I don't know what it is, but I'm sure there will be one. Thank you.